42. 1. Pathology, Introduction. Plant pathology, also known as phytopathology, is a branch of agricultural, biological or botanical science which deals with the study of diseases in plants, their causes, etiology, epidemiology, resulting losses and management. Plant pathology is related to many other sciences such as virology, mycology, bacteriology, microbiology, physiology, chemistry, genetics, biotechnology, etc., all of which provide the knowledge required for the correct diagnosis and management of plant diseases. About 34% of the crop produce is lost annually due to diseases, insect pests and weeds on the global basis, out of which 12% is lost due to diseases caused by fungi, bacteria or viruses, 11% due to nematodes, 7% due to insect pests and 3% due to weeds. When plant protection measures are not implemented, annual loss of 30 to 50% are common in major crops including horticulture. Late blight of potato caused by Phytophthora infestans was responsible for causing Irish famine in 1845 by destroying the potato crop, the staple food of the people. The population of Ireland was 8 million in 1840, which was reduced to 4 million after the famine. Brown spot of rice caused by Helminthosporium oryzae was responsible for Bengal famine in 1943, which many people think one of the reasons for the division of Bengal. Coffee rust caused by Hemilia vastatrix forced to cut down the coffee plants in Sri Lanka in 1867. In 1878, the downy mildew caused by Plasmopra viticola ultimately led to the discovery of Bordeaux mixture. Objectives of plant pathology: to study living, non-living and environmental causes of diseases or disorders of the plants. To study the mechanism of plant disease development. To study interaction between hosts susceptible and the pathogens. To develop systems of management of plant diseases and reducing losses caused by them. Definitions and concepts. Disease. Disease may be defined as a malfunctioning process that is caused by continuous irritation by a pathogen and or environmental factor resulting in some suffering producing symptoms. Disorder. The disease is caused by the deficiency of nutrients or unfavorable environmental are sometimes termed as disorders or physiological disorders. Pathogen. It is the agent responsible for inciting, pathos, i.e. ailment or damage. Literally, a pathogen is any agent which causes damage, but the term is generally used to denote living organisms, as fungi, bacteria. Most pathogens are also parasites as they derive the nutrients for growth from a living plant, the host or suscept. Parasite. These are the organisms which derive the food materials needed for their growth from other living organism, the host. All the pathogens are parasites, but all the parasites are not pathogens, as some of the parasites live on their hosts without causing any damage to them as 1644. Symbiotic relationships, E.g., Rhizobium bacterium in legume roots, mycorrhizae and lichens. Parasites may be obligate, biotrophs, if restricted to living tissues or facultative when they also colonize living or dead tissue. Necrotrophs grow on dead tissues which they kill in advance, thus more dangerous than biotrophs. Biotrophs are the organisms which, regardless of the ease with which they can be cultivated on artificial media, obtain their food from living tissues only in nature in which they complete their life cycle. They were earlier also called obligate parasites, E.g., rusts, smuts, powdery mildews, etc. Saprophytes. Saprobes are the organisms which derive their nutrition from the dead organic matter. Some parasites and saprophytes may have the faculty or ability to change their mode of nutrition. Facultative saprophytes are ordinarily parasites which can grow and reproduce on dead organic matter under certain circumstances. They are also called hemibiotrophs, which attack the living tissues in such a way as biotrophs but continue to grow and reproduce after the tissues is dead. A parasite is called necrotroph when it kills the host tissue in advance of penetration and then lives saprophytically, e.g. Sclerotium rolfsi and Pythium species. Similar to necrotrophs are facultative parasites which live as saprophytes, but under favorable conditions they can attack living plants and become parasites. The necrotrophs are also known as pathotrophs or pathophytes. Pathogenicity is the ability of a pathogen to cause disease under a given set of environmental conditions, whereas pathogenesis is the chain of events that leads to development of a disease in the host. It should not be confused with virulence and aggressiveness, used as a measure or degree or pathogenicity in a qualitative sense. Thus a pathogen may have several strains, races, of varying virulence, some of which may be avirulent, non-virulent. Parasitism is a phenomenon by which a plant parasite becomes intimately associated with the plant, it draws nutrition and multiplies and grows at the expense of the plant host. Virulence is a measure or degree of pathogenicity of an isolate or race of the pathogen. The term aggressiveness is often used to describe the capacity of a pathogen to invade and grow in the host plant and to reproduce on or in it. This term, like virulence, is used as measure of pathogenicity. Immunity of a plant against a disease is absolute quality. It denotes the freedom of plant from disease, when the pathogen cannot establish parasitic relationship with the host. High resistance and low susceptibility approach immunity. Disease resistance is the ability of an organism to overcome completely or in some degree the effect of a pathogen or other damaging factor, whereas susceptibility in the inability of the plant to resist the effect of the pathogen or other damaging factor. Hypersensitivity is the extreme degree of susceptibility in which there is rapid death of the cells in the vicinity of the invading pathogen. It halts the further progress of the pathogen. Thus, hypersensitivity is a sign of very high resistance approaching immunity. 1645. Infection is the establishment of the parasitic relationship between the pathogen and host following entry or penetration. Incubation period is the time elapsing between penetration and completion of infection, i.e., development of the disease symptoms. Invasion and colonization is the growth and multiplication of the pathogen through the tissue of the host varying extent. Inoculum is that portion of a pathogen which is transmitted to or contacts a host and is capable of infecting the host. Thus inoculum in bacteria are cells, in virus particle itself, in nematodes itself, eggs or cysts, in fungi, vegetative hyphae, spores. After inoculum has come in contact with a susceptible host, its ability to infect the host depends on several factors. One of such factors is the inoculum potential of the pathogen. The term coined by Horsfall, 1932, denotes the number of infective particles present in the environment of uninfected host. Epidemiology is mainly concerned with the development of epiphytotics, i.e. spread of pathogen in plant populations. However, in its wider sense, epidemiology includes most field aspects of disease, which involves interaction of population of crop plants, of pathogens and environment. Thus besides spread, modes of seasonal carry, over, and transmission of pathogen are also included under epidemiology. Important diseases of crop plants: crop disease, pathogen rice tundra, viral bacterial blight, wilt, Xanthomonas oryzae PV, oryzae blast, pyracularia grisae, P. oryzae, brown leaf spot, helminthosporium oryzae, sheath blight, rhizoctonia solni, false smut, ustilegenoides, V. rains, nematodes, pest nematodes, wheat rusts, Puccinia gramineae, FSP, Tritici, P. striformis F.sp, Tritici, P. recondita smut of wheat, Ustilego sigetum var, Tritici, U. Tritici. Kernal bunt telesia, Neovosea, indica folia blights, helminthosporium sativum and alternaria, triticina nematode pests, heterodera vene molia, corine bacterium, earcoccale, anguina, Tritici, pearl millet, downy mildew, sclerospora graminicola, ergot, claviceps fusiformis. 1650, general symptoms of plant diseases. Symptoms: external expression or the evidence of the abnormalities in the appearance of the diseased plants brought about by the pathogens after host-pathogen interaction. Sign: when the pathogen itself becomes visible on the host surface in the form of its organs or structures, sclerotia, mycelium, etc. Disease syndrome, a sum total of variety of symptoms produced by the disease. 1651, these enormous variety of symptoms basically belong to any of the three main categories, I. Necrosis, 2. Hypertrophy and hyperplasia, and 3. Hypoplasia. Necrosis. Necrosis is the commonest and most destructive type of effect. This kind of symptoms are produced by rather unspecialized parasites. Among these pathogens, the most destructive ones, Pythium, Botrytis, Fusarium causing wilts and many others, cause immediate and severe damage to host tissues and obtain their nutrients from cells killed in advance of colonization of host tissues by secretion of enzymes and toxins. They are thus called nectrophs, intermediate group between the necrotrophs killing host cells in advance and the specialized parasites as rusts, mildews, etc. do not kill cells in advance, but they invade and kill host cells. They kill host cells during their invasion. Such necrotrophs include fungi like Phytophthora, Venturi, Tuphrina and Calviceps. Necrosis caused by specialized parasites also like rusts, mildews, smuts, but only in later stages of attack when they sporulate. Thus during early stages of infection there is only slight adverse effect. Necrosis is also caused by bacteria and viruses. The various kinds of necrotic symptoms are as follows. Leaf spots, the cells are killed in a limited area to form lesion or spots. The spots are of various size and shape as zonate, target board, angular, frog eye. Tar spots are characteristic type of leaf spots which appear as raised, black-coated fungus bodies giving the appearance of a flat drop of tar on leaves. In some cases the dead tissue shrinks and separates from the healthy tissue. This condition is known as short hole. Streaks or stripes. These are elongated, narrow lesions on leaves, which are usually of brown shade. Examples are barely striped by helminthosporium. Blights. This gives a burnt appearance. There is sudden death of plant parts as leaves, blossoms, stamen, or twigs or even entire plant. The dead part usually turns brown or black and soon disintegrates. Examples are early and late blight of potatoes, some bacteria as Xanthomonas. Damping off. This is the condition where the stem is attacked near the ground level. The affected portion becomes constricted and weak, and finally disintegrates, causing the seedling to topple down and die. Damping off of vegetable seedlings is common example caused by Pythium and Phytophthora. Rots. The infected tissue dies, decomposes rapidly and turns brown. Fungi and bacteria which are able to dissolve cell walls cause this symptom. The rot may be root rot, leaf rot, stem rot, bud rot or fruit rot. Symptoms of plant diseases due to the character and appearance of visible pathogen, its structures and organs. I. Mildews. 1652. Mildews consist of white, grey, brownish or purplish pathogen growth on the host surface. Downy mildew is characterized by a tangled cottony or downy growth mostly on the lower surface of the leaves or other plant parts. Powdery mildew consists enormous number of spores are formed on superficial growth of the fungus giving a dusty or powdery appearance on the host surface. Black minute fruiting bodies may also develop in the powdery mass. Rust appears as relatively small pustules of the spores, usually breaking through the host epidermis. Pustule is a small blister-like elevation of the epidermis, often opening to expose spores. The pustules may be dusty or compact, and red, brown, yellow or black in color. 3. Smut means a sooty or charcoal-like powdery mass. The affected parts of the plants show black or purplish-black dusty areas. Symptoms usually appear on floral organs, particularly the ovulatory areas. The pustules on the leaves and stems are usually larger than those of rusts. 1653. Grain sites replaced by masses of teleospores. Mycelium follows. Mycelium invades growing point of the grain sites. Wheat plant teleospores land on flowers of healthy plants and infect developing grain. Mycelium invades young seedlings. Mycelium invades part of embryo in seed. 4. White blister-like pustules appear on the leaves and other parts of cruciferous plants which break open the epidermis and expose powdery masses of spores. Such symptoms are called white rust, although there is nothing common with them and the rusts. Blotch. It consists of superficial growth giving the affected plant parts i.e. fruits and leaves smoky, blotched appearance, e.g. sooty blotch of apple. Sooty blotch scab fly speck. 1654. 6. Sclerotia. A sclerotium is a compact, often hard mass of dormant fungus. Mycelium. Sclerotia are mostly dark in color and are found mixed with the healthy grains as in the case of ergot of wheat and rye. 7. Exudation. Mass of bacterial cells ooze out on the surface of the affected organs where they may be seen as a drop or smear in several bacterial diseases such as bacterial blight of paddy, gummosis of stone fruits and fire blight of apple and pear. They form crusts after drying. 8. Mycelial growth. Appearance of white cottony. Mycelial growth of the fungi like Dermatophora necatrix on affected roots of apple is an important diagnostic feature of white root rot in the field. 2. Symptoms resulting from internal disorders in the host plant's color change. Discoloration is change of color from normal. It is one of the most common symptoms of plant diseases. The green pigment of leaves disappears entirely and is replaced by yellow pigments. Etiolation is yellowing due to the lack of light. Chlorosis is yellowing due to low temperature, lack of iron, excess of the lime or alkali in soil and infection by viruses, fungi and bacteria. Albilinism is the phenomenon in which the leaves become devoid of any pigment and look bleached or white. Chromosis is change of color to red, purple or orange. Overgrowths or hypertrophy. Hypertrophy is the abnormal increase in the size of the plant organs due to increase in the size of the cells of a particular tissue, whereas hyperplasia is the abnormal increase in the size of the plant organs due to increase in the number of cells of which the tissue or organ is composed, owing to increased cell division. The overgrowths. Coys galls, curl, pockets or bladders, hairy root, witch's broom, intumescence etc. 3. Atrophy or hypoplasia or dwarfing. Atrophy is inhibition of growth and thereby showing stunting and dwarfing effect on the plants. The whole plant may be dwarfed or only certain organs are affected, e.g. rice dwarf, phony peach etc. 1654. 3. Necrosis. Death of the cells, tissues and organs occurs as a result of parasitic activity. The characteristic appearance of the dead areas differs with different hosts, host organs and with different parasites. Necrotic symptoms include spots, streaks or stripes, canker, blight, damping off, burn, scald or scorch and rot. Wilt characterized by drying of the entire plant. Leaves and other green or succulent parts lose their turgidity, become flaccid and droop down. Usually seen first in some of the leaves. Later, the young growing tip or the whole plant may dry up. May be caused by injury to the host system or the conducting vessels. Wilting due to disease is different from the physiological wilting where the plant recovers as soon as the supply of water is retained. Dieback or wither tip. Symptoms are characterized by drying of plant organs, especially stems or branches, from the tip backwards. It is also a form of necrosis caused directly by the pathogen or its toxins. 1655. Causes of plant diseases. Plant diseases are caused by a variety of pathogens. The word pathogen can be broadly defined as any agent or factor that incites, pathos, or disease in an organism. Thus in strict sense, the pathogens do not necessarily belong to living or animate groups. 1656. Abiotic, inanimate, factors. They include mainly the deficiency or excess of nutrients, light, moisture, aeration, abnormality in soil condition, atmospheric impurities etc. Examples are, black tip of mango, due to SO2 toxicity, Cara disease of rice, due to Zn deficiency, whiptail of cauliflower, Mo deficiency, hollow and black heart of potato, due to excessive accumulations of CO2 in storage, bitter pit of apple, due to Ca deficiency. Mesobiotic causes. These are the disease incidents which are neither living nor non-living. They are considered to be on the threshold of life. They are viruses. They are infections agents made up of one type of nucleic acid, RNA or DNA, enclosed in a protein coat. Examples of viral diseases of plants are potato leaf roll, leaf curl of tomato and chilies, and mosaic disease of many plants. Viroids. They are naked, infectious strands of nucleic acid. They cause diseases like potato spindle tuber, citrus ixocotis, chrysanthemum stunt, kadang of coconut palm, star crack of apple, etc. Biotic, animate, causes. This category includes the pathogens which are animate or living or cellular organisms. They are prokaryotes like bacteria which are unicellular prokaryotic microorganisms lacking true nucleus. Examples of diseases caused by true bacteria are brown rot or wilt of potato, soft rot of potato and vegetables, citrus canker, etc. Phytoplasma are Wallace prokaryotes and cause diseases like peach X. Fastidious bacterium, Xylella fastidiosa causes almond leaf scorch, Pierce's disease of grapevine. Eukaryotes are the organisms with true nucleus. Fungi, potato wart, Poderian disco mildew, rust, smuts, red rot of sugarcane. Nearly 80% of plant diseases are caused by fungi. Struminopiles, Oomycetes downy mildews, late blight of potato, white rust of crucifers, damping of etc. Protozoa, heart rot of coconut palm and phloem necrosis of coffee. Algae, red rust of mango or papaya or lychee metazone animals, nematodes root knot of vegetables, ear cockle of wheat, citrus decline etc. 6. Parasitic flowering plants, phanerogamic plant parasites dodder, striga, orobranch, loranthus, phodendron etc. 1705. 2. Classification of disease. The nature and concept of disease is very much varies. A number of definitions have been proposed for a diseased plant. Anon, 1950, defined the disease as harmful deviation from normal functioning of physiological processes. According to Stuckman and Harrar, 1957, a plant disease is a physiological disorder or structural abnormality that is deleterious to the plant or any of its parts or products or that reduces their economic value. Disease is a malfunctioning process that is caused by continuous irritation. Components of disease pyramid. It is common to recognize three important components of disease: a susceptible host, a virulent and aggressive pathogen, and a favorable environment. These three components constitute the traditional disease triangle. Stevans, 1960, Jadokes, 1972, and Van der Planck, 1975, however also recognized a fourth factor, time, as an equally important component of plant disease. Environment. Pathogen plant. Time is important in many ways as the time of year when host and pathogen are juxtaposed, time of year when duration of leaf wetness while temperature is favorable, time of the next period favorable for spore dispersal and germination and for establishment of infection etc. The effect of time on disease development becomes apparent when we consider the importance of time of year, that is, the climatic conditions and stage of growth when host and pathogen may coexist, the duration and frequency of favorable temperature and rains, the time of appearance of the vector, the duration of the cycle of a particular disease, the earliness and lateness of maturity of host etc. Adding time to the disease triangle as a fourth component results into a disease pyramid. 1936. If the four components of disease pyramid could be quantified, its volume would be proportional to the amount of disease on a plant or in plant population, and the area under a disease progress curve. Reducing any component of disease pyramid through management, the host susceptibility, the virulence of pathogen, degree of favorableness of time or of the environment, will decrease the amount of disease in a plant or the intensity of the epidemic in a population of plant and thence the disease volume development of pyramid. In plant cultivated pathologist thus iron can greatly manage influenced disease by components. A fifth component, in many humans ways, plants is also. Humans affect disease development in various ways. They affect the kind of plants grown in a given area, their level of resistance, the number of plants planted, time of planting, and density of plants, by their cultural practices, chemical and biological control etc. Humans affect the amount of primary and secondary inoculum available to attack plants. They also modify the ways, manipulating the time of planting and harvesting etc. The human component that directly and indirectly affects the disease development. In the figure, host, pathogen and environment are each represented by one of the sides of the triangle. Time is represented by one of the sides of the line arising from the center of the triangle and humans as the peak of the pyramid whose base is the triangle and height is the length of time. In this way humans interact with and influence each of the other four components of the disease. Classification of diseases. Plant diseases may be grouped into various types. I on the basis of their method of perpetuation and mode of primary infection. 1. Soil-borne diseases. In pathogens causing diseases like pre-emergence killing and damping off of seedlings, seedling blight, Pythium, Phytophthora, Fusarium, root rots, Verticillium, Caletotrichum, and vascular wilts, Fusarium and Verticillium, the primary infection takes place from the soil. These soil-borne pathogens survive in soil or on infested plant debris lying in soil either as their resting spores or as mycelial strands and rhizomorphs. They all attack the root systems of host plants. Under favorable conditions the spores germinate and infect roots either at seedling stage or an adult plant. Airborne diseases. Some pathogens however, infect the host plant through air and bring primary as well as secondary infection. Fungi as rusts and mildews develop their thick-walled resting structures, teleospores in rusts, Calistodicea in powdery mildews, or spores in downy mildews. Under favorable conditions these germinate to produce thin-walled spores. The spores are disseminated by wind and infect the shoot of the plant. Loose smuts bring about secondary infection through air. Seed-borne diseases. 1937, some pathogens survive as dormant mycelium in the seeds or other propagative structures of host plants. In most smuts, the dormant mycelium becomes active at the time of seed germination. The mycelium grows with the growing apex of the seedlings. Later the mycelium spreads throughout the entire plant including the floral parts. 2. On the basis of their extent of occurrence and geographic distribution. 1. Endemic diseases. A disease is classified as endemic when it is constantly present in a moderate or severe form and is confined to a particular country or region. E.g. wart disease of potato, Syncytium endobioticum, is endemic in Darjeeling. Epidemic or epiphytotic diseases. An epidemic disease usually occurs widely but periodically in a destructive form. The pathogen may be present, as in endemics, but the environmental factors favorable for rapid disease development occurs only periodically. E.g. cereal rusts and powdery mildew. Sporadic diseases. These diseases occur at very irregular intervals and locations and in relatively fewer instances. E.g. angular leaf spot and blotch disease of cucumber. Pandemic disease. Pandemic outbreak is an epidemic which occurs over vast and extensive areas causing severe losses. These occur all over the world and result in mass mortality. E.g. late blight of potato. Procedure of disease diagnosis. In plant disease diagnosis, it is first necessary to determine whether the disease is caused by a pathogen or an environmental factor. On the basis of long experience, one may, in few cases of very typical symptoms of disease present, decide whether the cause is a pathogen or an environmental factor. To know the real cause pathogen environmental factor, the common practice is to compare the symptoms observed with standard literature, books, charts, etc., describing the characteristic symptoms to known diseases on specific plants. American Phytopathological Society has published the Compendia series of disease symptoms. In most cases, however, one has to examine the symptoms in detail for correct diagnosis. Infectious diseases. In diseases caused by fungi, bacteria, parasitic higher plants, nematodes, viruses, mycoplasmas and protozoa, a few or large number of these pathogens may be present on the surface of plants or inside the plants. 1941, their presence on or in the plant indicates them as possible cause of the disease. Some of these can be detected, with experience by using magnifying lens or the naked eye, as nematodes higher parasitic plants, some fungi. More frequently, one has to make detailed microscopic examination for correct identification, fungi, bacteria, nematodes. If no such pathogens are observed on plant surface, one has to look for additional symptoms and scan the internal tissues of attacked plant for pathogens inside the plant, vascular elements, roots, stem base, leaf, etc. Parasitic higher plants can be easily detected simply by their presence on a plant. For nematodes, if the nematode is present on, in, or in the rhizosphere of plant showing some type of symptoms, the nematode may be the pathogen. It can be identified to generic and species level. One has to follow a relatively longer procedure for diseases caused by fungi and bacteria. When mycelia and spores of fungi or bacteria are present on the diseased area, these may be the real cause of disease, or these may be saprophytes living on dead plant tissues, killed by some other cause. For fungi, one has to determine the exact nature first after making microscopic studies for the morphology of mycelium, fruit bodies, spores, etc. The fungus can be identified with the help of authentic literature. We have to compare the symptoms and the detailed morphological characters of the fungus with the diseases already known and described in the literature for correct identification. If no such fungus is known to cause a disease, especially the one with symptoms similar to the ones under study, then the fungus observed should simply be a saprophyte. For bacteria, identification of the pathogen is based primarily on the symptoms, the constant presence of large numbers of bacteria in the affected area, and the absence of other pathogens. Selective media are available for the selective cultivation of plant pathogenic bacteria. The bacterial pathogens are identified on cultural and nutritional characteristics. After obtaining in pure culture, a single colony is used for reinoculation of susceptible host plant and reproduction of the symptoms. These symptoms are then compared with those produced by known species of bacteria. A range of techniques including ELISA have been used to detect and identify plant pathogenic bacteria. Diseases caused by molecules are diagnosed on the basis of symptoms, graft transmissibility, insect transmission, electron microscopy, sensitivity to tetracycline antibiotics but not to penicillin, serodiagnosis, and sensitivity to moderately high, 32-35, temperatures. They produce characteristic types of symptoms: stunting of plants, yellowing or reddening of leaves, proliferation of shoot and root, production of abnormal flowers, eventual decline and death of plant. Molecules are small polymorphic, wall, less bacteria, visible under electron microscope only living in the young phloem cell of points. For viruses and viroids, we should observe the symptoms first, as their symptoms are quite distinct and disease can be easily identified on their basis. If it is not possible, the viruses are identified as follows. 1941. Transmission tests to specific host plants by sap inoculation or by grafting and in some by vectors. For those in which specific antisera are available, one may use ELISA, gel, diffusion, microprecipitation, fluorescent antibody staining techniques, W electron microscopy, microscopic examination of infected cells, electrophoretic tests, particularly for viroids and viral acid, and DNA hybridization. Non-infectious diseases. If no pathogen is detected, cultured or transmitted from a diseased plant, the disease should be presumed to be due to an abiotic agent, i.e. environmental factor. A number of environmental factors cause several physiological disorders from plants. The specific factor responsible for the disorder may be determined by observing changes in the environment. Some factors cause specific symptoms on plants that help determine the cause of disorder, but for most of them one has to look for details environmental conditions where the plant's growing. Coates postulates for identification of a previously unknown disease we have to follow some sequential steps to ascertain that the isolated organism is the real cause of the disease. Even if there are no previous records of the organism isolated from or associated with plants, its mere presence does not prove that it is the real cause of the disease. During the late 19th century, a bacteriologist called Robert Koch laid down a set of rules for confirming that an organism is the cause of a disease. These are now known as Koch's postulates. The following steps are taken in order to prove the pathogenicity of the organism associated with the disease. The pathogen must be found associated with the disease in all the diseased plants examined. The pathogen must be isolated and grown in pure culture on nutrient media and its characteristics described, if one, specialized necrotrophic parasite, or it must be grown on a susceptible host plant, if specialized biotrophic pathogen, and its appearance and effects recorded. The pathogen from pure culture must be inoculated on healthy plants of the same species or variety on which the disease appears, and it must produce the same disease with identical symptoms on the inoculated plants. The pathogen must be isolated in pure culture again from the affected part of the inoculated plant, and its characteristics must be exactly like those observed in step 2. If all the above steps, usually known as Koch's postulates, have been followed and proved true, then the isolated pathogen is identified as responsible for the disease. It is possible to implement Koch's postulates easily with fungi, bacteria, parasitic higher plants, nematodes, some viruses, some viroids, and the spiroplasmas, which can be isolated, grown as pure culture, or purified, and could then be introduced into plants. 1942. For some other pathogens, such as some viruses, fastidious, phloem, inhabiting bacteria phytoplasmas, and protozoa, this may be tedious due to problems in their culture or purification. Red blood cell. Causative agent absent. Healthy organism suspected agent. Causative agent. Causative agent. Organisms. Organism. 1942. 3. Plant disease epidemiology and forecasting. Epidemiology deals with outbreaks and spread of disease in a plant population. When a pathogen spreads to and affects many individuals within a population over relatively a large area and within a relatively a large area and within a relatively short time, the phenomenon is called an epidemic. An epidemic has been defined as any increase of disease in a population, or the dynamics of change in plant disease in time and space. A plant disease epidemic, or epiphytotic, implies the development and rapid spread of a disease on a particular kind of crop plant cultivated over a large area, a large field, a valley, a section of a country, the whole country, or even a part of continent. The study of epidemics and of the factors that influence them is called epidemiology. Some plant disease epidemics, for example, wheat rusts from time to time in many countries of the world, and grape downy mildew in USA and France, have caused tremendous losses of produce over large areas. Others, like chestnut blight, Dutch elm disease and coffee rust, have threatened to eliminate certain plant species from entire continents. Still others have caused untold sufferings to humans. The Irish famine of 1845 to 1846 caused by the late blight epidemic of potato and the Bengal famine of 1943 caused by brown spot epidemic of rice are related to the history of mankind. In epidemics, though host and environmental factors are equally important components, rate of multiplication of the pathogen also determines its capacity to spread a disease in plant population. Under favorable conditions, pathogens multiply and spread through populations of susceptible plants, sometimes causing extensive outbreaks of disease. On the basis of the mode of multiplication of pathogen, the diseases may be of two types, simple interest disease and compound interest disease. Epidemiologically, the diseases have been described as simple interest disease. In these diseases, the rate of increase is mathematically analogous to the simple interest in money. There is only one generation of the pathogen in the life of the infected crop. The primary inoculum is seed or soil-borne and secondary infection is rare. All the infections noticed in the field are from the pre-existing inoculum in the soil. Most important example is loose smut of wheat, Eucritiaceae, where the inoculum is internally seed-borne or is carried in the seed, resulting in the infected ears in the season showing black powdery loose mass in place of grains. In wilt and root rot diseases, the primary inoculum is important and there are very remote chances of secondary spread, even if the pathogen sporulates, due to the soil barriers and other factors. Compound interest disease. In these diseases, the rate of increase is mathematically analogous to compound interest in money. 1943. The pathogen produces spores at a very rapid rate which are disseminated by external agencies like air, thereby infecting other plants. The incubation and sporulation period is also very short. The infection cycle is repeated many times during the cropping season. Secondary inoculum plays a vital role in the development of epidemics in such diseases. The prevailing environmental conditions play an important role in such diseases. Wheat rust, late blight of potato and apple scab are some diseases of this type. Epidemics of plant diseases are also designated as epiphytotics. Pandemic are destructive epidemics developing on a continental scale. It is difficult to say at what point an outbreak of disease becomes an epidemic or even to define it. On the basis of the relative time taken by it to develop, an epidemic may be slow epidemic or rapid epidemic. Elements of an epidemic. Plant disease epidemics develop as a result of the timely combination of the same elements that result in disease: susceptible host plant, virulent pathogen, and favorable environmental conditions over a fairly long period of time. Humans many unwittingly help initiate and develop epidemics through some of their activities. Very often, however, humans stop the initiation and development of epidemics by using appropriate control measures under situations in which epidemics would certainly occur without such human intervention. Thus, the chance of an epidemic increases when the susceptibility of the host plant and virulence of the pathogen are higher, as the environmental conditions approach the optimum level for the growth, reproduction and spread of the pathogen, and as the duration of all favorable combination is prolonged or repeated during the growing season of the crop plant. To consider the interactions between the various components of disease epidemic, the disease triangle, discussed earlier to describe the interactions of the components of disease. It was indicated that the disease triangle can be expanded to include two more components, time and humans. In fact, the amount of each of the three components of disease and their interaction in the development of disease are affected by a fourth component, time. Both the specific point in time at which a particular event in disease development occurs and the length of time during which the event takes place affect the amount of disease. Thus adding time component to the disease triangle results into a tetrahedron or pyramid, in which each plane represents one of the components. Such a disease tetrahedron, pyramid, describes the epidemic development. The effect of time on disease development becomes apparent when we consider the importance of year, that is, the climatic conditions, and stage of growth when host and pathogen may coexist, the duration and frequency of favorable temperature and rains, the time of appearance of the vector or the duration of the cycle of a particular disease. If the four components 1943 of disease pyramid could be quantified, its volume would be proportional to the amount of disease on a plant or in plant population. Disease development in cultivated plants is also greatly influenced by a fifth component, humans. Humans affect disease development in various ways. They affect the kind of plants grown in a given area, their level of resistance, the number of plants planted, time of planting, and density of plants. By their cultural practices, chemical and biological control etc. humans affect the amount of primary and secondary inoculum available to attack plants. They also modify the environment on disease development in several ways, manipulating the time of planting and harvesting etc. The human component has sometimes been used in place of the time component in disease pyramid, but it should be considered a distinct fifth component that directly and indirectly affects the disease development. Factors affecting development of epidemic. Host, pathogen, environment and human activities are the factors affecting development of an epidemic. Host factors. These include external as well as internal factors of host plants. These are as follows. Levels of genetic resistance or susceptibility. Host plants with high levels of vertical resistance do not allow a pathogen to become established unless a new race of pathogen develops. Plants with lower levels of horizontal resistance become infected. Degree of genetic uniformity of plants. When genetically uniform host plants, particularly in terms of genes for disease resistance, are grown over large area, there is much possibility of the appearance of a new race of pathogen to attack them, leading to an epidemic. This is the reason of the highest rates of epidemics generally on vegetatively propagated crops, with next highest in self-pollinated crops, whereas the lowest in cross-pollinated crops. Type of crop and age of plants. These are also important in epidemic development. In annual crops like corn, wheat, vegetables, cotton, tobacco etc. epidemics generally develop much more rapidly, in few weeks, than in the perennial woody fruit and forest trees, slow epiphytotics, some taking several years. In pear decline, Dutch elm disease, chestnut blight, epidemics are very slow. Plants' susceptibility to disease also changes with age, and generally three trends of disease progress could be seen. In some, such as pythium damping of and root rots, downy mildews, leaf curl of peach, rusts, systemic smuts, bacterial blights and viral infections, the hosts, or their parts, are susceptible only during the growth period, becoming resistant when adult resistance. With several diseases like rusts and viral infections, plant parts are resistant again before they are fully expanded. In other diseases like blossoms or fruit infections by botrytis, penicillium, monilinia and glomerella, and in all post-harvest diseases, plant parts i.e. fruits are resistant during growth and early adult period but become susceptible near ripening. 1946. Still in diseases like late blight of potato, early blight of potato and tomato etc., there is a stage of juvenile susceptibility during the growth period of plant, followed by a period of relative resistance in the early adult period and then susceptibility after maturity. Pathogen factors. These include the following. Virulence level. Virulent pathogens infect host rapidly and produce large amounts of inoculum at faster rates. Inoculum density. The greater the amount of inoculum near the host, within fields, more inoculum reaches the plants, and at an earlier time. Reproduction pattern of pathogen. Pathogens, such as most fungi, bacteria and produce many generations in a single growing season. These are polycyclic pathogens that usually cause rusts, mildews and leaf spots and are responsible for sudden catastrophic disease epidemics. Such pathogens are high birth rate pathogens, causing rapid epidemics as the diseases spread in compound interest manner, compound interest diseases. Some soil fungi, as Fusarium spp, Verticillium spp and most nematodes usually have 1 to 4 reproductive cycles per season. Limitation in their dispersal limits their potential to cause sudden epidemics in a single season. They often cause more localized, slower epidemics. There are pathogens like smuts and several short-cycled rusts that lack a repeating spore. They require the entire year to complete life cycle, monocyclic pathogens. In such cases, inoculum builds up from one year to the next, and the epidemics develop over several years. Similarly, epidemics caused by pathogens that require more than 1 year to complete a reproduction cycle are slow to develop, slow epidemic. They are low death rate pathogens and cause simple interest diseases. Some examples are cedar, apple rust, 2 years, and white pine blister rust, 3 to 6 years. Ecology of pathogen. As in most fungi and parasitic phanerogams, inoculum is produced on the surfaces of aerial parts of plant. This can be dispersed easily to longer distances and can cause widespread epidemics. Other pathogens like vascular pathogens, fungi as well as bacteria, mycoplasmas, viruses etc. reproduce inside the plants. The inoculum in such cases can spread only with the help of vectors. Thus epidemic development is possible only when there are abundant and active vectors. In soil-borne fungi, bacteria and nematodes, which produce inoculant plant debris lying in soil, inoculum dispersal is very slow. Mode of spread of pathogen. Inocula of rusts, mildews, leaf spot fungi are spores that can be easily dispersed by air and strong winds over long distance. They cause most frequent and most widespread epidemics. The next important group is of those whose inocula are carried by airborne vectors. These are many viruses. Windblown rain pathogens, such as anthracnose and apple scab fungi and most bacteria, cause every year severe but localized epidemics within a field, a growing belt, etc. Seed and soil-borne pathogens suffer from several limitations and hardly cause sudden or widespread epidemics but may cause local slow-spreading diseases. 1946. Environmental factors. The concurrent presence in the same area of susceptible plants and virulent pathogens does not always guarantee numerous infections and the development of an epidemic. That's how the environment has a controlling influence on development of epidemics. The environment may affect each component of the epidemic, i.e. host as well as pathogen, vectors, etc. The most important environmental factors that affect disease epidemics are moisture, temperature, and the activities of humans in terms cultural practices and control measures. We have already considered the role of these individual factors in the beginning of this chapter. Development of an epidemic. For a disease to become significant in a field, and particularly if it is to spread over a large area and develop into a severe epidemic, the right combinations of environmental factors must occur either constantly or repeatedly and at frequent intervals over a large area. Even in a single, small field that contains the pathogen, plants almost never become severely diseased from just one set of favorable environmental conditions. It takes repeated infection cycles and considerable time before a pathogen produces abundant inoculum to cause economically severe epidemics in the field. Once large populations of the pathogen are available, they can attack, spread to nearby fields and cause a severe epidemic in very short time, many be in a few days. A plant epidemic can occur in a garden, a greenhouse or a small field, but epidemic generally implies the development and rapid spread of a pathogen on a particular kind of crop plant cultivated over a large area, such as large field, a valley, a part of the country, entire country or even part of a continent. Therefore, the first component of plant disease epidemic is a large area planted to a genetically uniform crop plant, with the plants and the fields being close together. The second component of an epidemic is the presence or appearance of a virulent pathogen. Such cohabitations of host plants and pathogens occur, of course, daily in numerous locations. Most of these, however, cause only local disease of varying severity, destroy crop plants only to a limited extent, and do not develop into epidemics. Epidemics develop only when the combinations and progression of the right sets of conditions occur. These include appropriate temperature, moisture, and wind or insect vector, coinciding with the production, spread, inoculation, penetration, infection and reproduction of the pathogen. The third component of the epidemic is this combination of right sets of environmental conditions at appropriate stage of the host plant and the pathogen. Thus, for an epidemic to develop, the small amount of original, primary inoculum of the pathogen must be carried by wind or vector to some of the crop plants as soon as they begin to become susceptible to that pathogen. The moisture and temperature must temperate for germination and subsequent infection to take place. After infection, temperature and or other conditions must be favorable for rapid growth and reproduction of the pathogen, short incubation period and infection cycle, so that numerous new spores are available as quickly as possible. The 1947. Moisture, rain, fog, dew, then must be enough for abundant release of spores. Winds of proper humidity and velocity, blowing towards the susceptible crop plants must prevail during this time. Lastly, and the most important fourth component of an epidemic is that in each new location, however, the same set of favorable moisture, temperature, wind or vector conditions must be repeated so that infection, reproduction and dispersal of the pathogen can occur as quickly as possible. Furthermore, these conditions must be repeated several times within each location so that the pathogen can multiply, increasing the number of infections on new plants. These repeated infections usually result in the destruction of almost every plant within the area of an epidemic. Fortunately, the most favorable combinations of conditions for disease development do not occur very often over very large areas. Therefore, spectacular plant disease epidemics that destroy crops over large areas are relatively rare in nature. However, small epidemics involving plants in a field or a valley occur quite frequently. With diseases like cereal rusts, late blight of potato, rice blast, strips disease of barley, the environmental conditions seem usually to be favorable, and small disease epidemics would occur every year in absence of adequate control measures, chemical sprays, resistant varieties, etc. Briefly, we may say that following conditions favor the epiphytotic, I, virulent pathogen with short sporulation, latent, period, 2, rapid spore germination and infection, 3, susceptible plant at susceptible stage of growth, 4, intensive monoculture of susceptible plants over large areas, and V, environmental conditions favorable to the pathogen at all points in the infections cycle, i.e., sporulation and subsequent liberation, dissemination and deposition of spores in variable condition, germination, infection and growth in host tissues. Whether during the intercrop period as well as during crop season can be important in seasonal carry over, and it may affect the initial outbreak and subsequent spread of the pathogens. Modeling and computer simulation of epidemics. An epidemic is a dynamic process. It takes birth on one or a few plants and then, depending upon the coincidence of right sets of host, pathogen and environmental conditions, increases in severity and spreads over a large geographic area until it finally dies down. It may come to a stop when all host plants are killed, become resistant to pathogen, or they age or are harvested. In many cases, epidemics slow down or come to a stop when weather turns unfavorable. Epidemics, thus in many respects are like hurricanes. In crop production, we thus become interested to know all such elements and conditions which initiate epidemics and increase their severity. In order to predict the size, path, pattern and time of appearance of epidemics there are used suitable observations, measurements, mathematical formulae and computers. Each plant epidemic, say late blight of potato, stem leaf rust of wheat, apple scab or dowry powdery mildews of grape etc. follows a predictable course in each location each year. The course of epidemic varies with the host varieties and pathogen races present, amount of primary inoculum in the beginning and levels of moisture and temperature ranges during the epidemic. More we know of all such components, better we can understand and describe the epidemic, and 1948. Better we can predict the severity and direction of an epidemic, more comfortably we are in a position to take appropriate control measures. In an effort to improve our ability to understand and predict the development of an epidemic, plant pathologists since 1960s have been developing models of potential epidemics of the most common and serious plant diseases. The construction of a model takes into account all the components of a specific plant disease for which there is information for quantitative treatment i.e. mathematical formulae. The models so constructed in fact are crude simplifications of real epidemics like a toy car. In modeling plant disease epidemics, each component and subcomponent of the epidemic may be considered equivalent to one of the parts of toy model. When we have enough information about the values of the subcomponents of an epidemic at different stages and under mathematical equation or equations, a mathematical model, that describes the epidemic. The availability of computers has allowed plant pathologists to write programs that allow simulation of epidemics of several plant diseases. Several such programs have been written in developed countries like USA and Europe. One of the first computer simulation programs, called Epidem, was written in 1969, to stimulate early blight epidemics of tomato and potato caused by alternaria. Thereafter, computer simulators were written for Cercospora blight of celery, Cercos, for Mycosphaerella blight of chrysanthemums, Mycos, for southern corn leaf blight, caused by Cochilobolus maedis, Epicorn, and for apple scab caused by Venturi iniquilis, Epivin. A more general simulator, called Epidemic, though designed for stripe rust of wheat, could also be modified for other diseases. Such simulators are now available for several other diseases also. In a computer simulation of an epidemic, the computer is fed with the data on various subcomponents of epidemic along with the control measures at specific points in time. The computer then provides continuous information not only on the spread and severity of the disease over time, but also final crop and economic losses likely to be caused. Computer simulation is very useful to plant pathologists as well as farmers. Forecasting of plant disease epidemics. Forecasting is in fact applied epidemiology. Plant disease forecasting and monitoring provides early information about the probable occurrence of a disease to facilitate chemical prophylaxis at appropriate time either to stop pathogen multiplication or further spread of the disease. Early information is essential to determine number of sprays and schedules to make economically sound disease control, and limit the chance of development of pathogen resistance to the pesticides. Forecasting is extremely useful to farmers in practical management of crop diseases. Disease forecasting allows the prediction of probable outbreaks or increases in intensity of disease, and therefore, allows us to determine whether, when, and where a particular management practice should be applied. In managing the diseases of their crops, farmers must always weigh the risks, costs, and benefits of numerous decisions. For example, besides so many other things relevant to cultural practices, they most frequently need the forecasts that will help them determine whether a plant infection is likely to occur so that they can decide whether to spray a crop right away or wait for more days before spray. If disease forecasting allows them to wait, they can reduce the 1948 expenditure on chemicals etc. and on labor. Maybe, this operation is not required at all, which would save money substantially. Forewarning is an essential component of IPM. In India, the efforts on pest forewarning have been put in application for field use in crops like potato and grapes. Attempts of forewarning of major rice pests for a few locations have been made possible based on historical data sets and specific field studies during the last decade through National Agricultural Technology and National Agricultural Innovation (NAP and NIAP) projects. The e-pest surveillance done under the National Innovations in Climate Resilient Agriculture (NICRA) by NSIP has sustained and widened the scope of forewarning. Indian Agricultural Statistics Research Institute (IASRI) has played a prominent role in developing methodologies for forewarning of different aspects relating to alternaria blight, white rust, powdery mildew and aphid, mustard, aphid, potato, American bollworm, pink bollworm, spotted bollworm and whitefly, cotton, Spodopter litura, late leaf blast and rust, groundnut, pyrilla, early shoot borer and top borer, sugarcane, podfly, pod borer, sterility mosaic and phytophthora blight, pigeon pea, fruit fly, hopper and powdery mildew, mango, and gallmage, rice, for selected centers. Two types of forecasting, short-term and long-term, are possible if sufficient data on weather, host and pathogen are available. Prediction of disease during the crop season is called short-term forecasting while the prediction of disease made years in advance is called long-term forecasting. Advantages. 1. Forecasting is extremely useful to farmers in the practical management of crop pests and diseases. 2. Forecasting allows the prediction of probable outbreaks or increases in intensity of disease, and therefore allows us to determine whether, when and where a particular management practice should be applied. 3. Forecasting helps the farmers to determine whether a plant infection is likely to occur so that they can decide whether to spray a crop right away or wait for more days before spray. If disease forecasting allows them to wait, they can reduce the expenditure on chemicals, etc., and on labor. Maybe, this operation is not required at all, which would save money substantially. E.G. The models developed for mustard aphid have been used by National Research Center for Rapeseed Mustard, Bharatpur to provide forewarning to the farmers consecutively for 3 years, which enabled them to optimize plant protection measures and save expenditure on unnecessary spray of chemicals. Requirements or conditions of plant disease forecasting. The disease must be causing economically significant damage in terms of loss of quantity and quality of the produce in the area concerned. The onset, speed of spread and destructiveness of the disease is variable, mostly due to dependence on the weather, which is variable. Control measures are known and can be economically applied by the farmer when told to so. 1949. Information on weather disease relationship is fully known. Basis of forecasting or methods of forecasting plant diseases. Forecasting is predicted in different ways for different kinds of the pathogens and the weather factors that affect disease development. Disease forecasts are based on the following factors. Plant disease forecasting has been applied in many diseases on the basis of weather conditions during the intercrop period, weather during crop season, amount of disease in the young crop, amount of inoculum in the air, soil or planting material. Forecasts based on weather conditions during intercrop period and amount of primary inoculum. Weather during intercrop period is closely related to the survival of many plant pathogens, mostly through the severe cold of winter months and in some cases to that of pathogen vectors also. Erwinia stuartill, causing Stuart wilt of corn, survives the winter in the bodies of flea beetle sets vectors. An assessment of vector population at the onset of spring gives an indication of the extent of survival of the vector through the cold months. Where the sum of mean temperatures for 3 winter months of December, January and February at a given location is less than 1, c, the most of vectors die and therefore no serious disease is expected. In contrast, mild winter permits good survival of vectors leading to severe disease outbreak. For curly top disease of sugar beet, severity of disease outbreak is correlated with the number of vectors that successfully overwinter. In California, USA, fire blight of apple and pear incited by Erwinia amylovora became severe if the daily average temperature exceeded a disease prediction line, obtained by drawing a line from 16.7 C on March 1 to 14.4 C on May 1. This pathogen multiplies very slowly at temperatures below 15 C, and this makes the initial inoculum inadequate for any strong attack. Some fungal pathogens affecting temperate region crops like those causing apple scab, brown rot, ergot of rye, etc. survive winter by developing resistant structures. The forecasters look for them to get hints about their prospects in the coming season. For some soil-borne fungal pathogens like verticillium and sclerotium spp and cyst nematodes heterodera and globodera spp, greater the amount of propagules, sclerotia and cysts, respectively, more severe is the disease. Weather conditions during the crop season and the production of secondary inoculum. The temperature and moisture levels are very critical during the crop season for the development and spread of some airborne diseases. 1950. Severe outbreaks are likely to occur if certain combinations of temperature and moisture levels are available for a certain period of time. This requirement is different for different diseases. Several leaf spot diseases of fungal origin, for example tick disease of groundnut, Tersicum blight of corn, apple scab and paddy blast, can be predicated by taking into account the number of spores trapped daily over the cultivated field, the temperature and relative humidity over a certain period of time. Amount of disease in the young crop. Extent of disease developed in the young crop may occasionally provide a reliable indication of the likely severe development of the disease in the mature crop. E.G. Leaf rust of wheat. The young crop mostly gets infected from overwintering local infections. The amount of infection at onset of spring often determines the subsequent development of this disease, as the weather following is generally favorable. The crop is, therefore, periodically assessed to keep track of the infection and control measures are recommended accordingly. Amount of inoculum in soil, planting material and air. In many diseases, the primary inoculum comes from last year's infected crop residues lying in the field. Overwintering fungal pathogens produce spores at the onset of the growing season which function as primary inoculum and initiate initial infections in some plants of the new crop. Examples are apple scab, brown rot of stone fruits or ergot of rye. The amount of such residues lying in the field or the plantation floor gives an indication of the availability of inoculum at the start of the season, and if the level is high, a forecast can be made. Some successful examples of plant disease forecasting. 1. Late blight of potato. Holland pioneered the development of forecasting and spray warning services for the control of late blight of potato. Van Everdingen, 1926, analyzed the combined effect of several weather conditions on the development of Phytophthora infestans and evolved 4 rules, popularly known as Dutch rules, on which the appearance of blight was observed to depend. I. Night temperature below dew point at least for 4 hours. 2. A minimum temperature of 100 C or above. 3. A mean cloudiness on the next day of 0.8 or more. And 4. At least 0.1 mm rainfall during the next 24 hours. When all these 4 conditions were obtained in Holland, potato blight was expected after the next 7 days, and therefore the control measures were immediately recommended to the farmers. 1950. 2. Apple scab forecasting in Himachal Pradesh. In Himachal Pradesh, when the apple scab appeared in epidemic form in late 1970s onward, 5 apple scab monitoring and research laboratories were established at Mashobra, Kotai, Thanegar and Sarahan in Shimla, and Bajora in Kullu district. Apple scab infection periods were predicted by using equipments like DeWitt 7-day leaf wetness recorder, Reuters-Stokes Apple scab predictor, and SWG biomat at different locations. When the conditions congenial for the development of Apple scab were recorded, this information was given to the farmers by blowing sirens, radio broadcasts, and personal contacts. Of all the systems, Reuters-Stokes predictor was found to be the most efficient in predicting the infection periods at Kotai. Different fungicides could be effectively used depending on their after-infection, curative, pre-symptom or post-symptom, eradicative activities. This is regarded as the best example of plant disease forecasting in India till date. Rice blast. In India, forecasting rice blast, Pyracularia ozizae, is done by correlative information method. It is predicted on the basis of minimum night temperature 20 to 26 C in association with high relative humidity of 90% or above. Computer-based forecasting system has also been developed for rice blast in India. For wheat stem rust. Forecasting wheat stem rust epidemic is done by analyzing their in samples which give precise data for inoculum present in the air. Moreover, several wind trajectories are also prepared to survey the airborne primary inoculums and its deposition. It has been observed that primary inoculum comes from South India to the plains of Central and North India. Amount of inoculum in soil, planting material and air. In the seed-borne plant diseases, the extent of infection and contamination in the seeds or planting material can be easily estimated in the laboratory. This can be useful for predicting seed-borne smut, bacterial and viral diseases. Depending on the level of infection or contamination, the seed or planting material can be rejected outrightly or made free from the pathogens by chemical or thermal treatments. Some soil-borne fungal pathogens may survive in soil as hyphae, but most of them survive in soil as resting structures like sclerotia, chlamydospores and strumtha. The soil-borne inoculum can be approximately determined, and if exceeds certain limits, susceptible variety may not be grown in such fields. 1951. Blight and root rots caused by Sclerotium rolfsi are good examples. Challenges. In developed countries, pesticide use is warranted by well-established monitoring, surveillance and forecasting system. It is generally done by established relationships between pathogen population and physical weather parameters like air temperature, rainfall, relative humidity, cloudiness, dry wetness or leaf wetness duration. Field monitoring for pre-disease symptoms at susceptible stage and monitoring of favorable weather conditions make the basis of disease monitoring. Forecasting and monitoring of major airborne diseases have been relying on the knowledge on biology and ecology of the pathogen, quantitative seasonal studies over several years, season range, and variation in the population pattern, geographical distribution and weather records. Forecasting systems for potato late blight, apple scab, powdery mildew of mango, beer and rice blast are now available. However, forecasting systems developed elsewhere could be adopted with local situation after expert judgments and field trials. But for practical utility in growers' field there is still a lacuna. In this connection, training program for the plant pathologists, extension persons as well as for the grower is necessary. New tools in epidemiology. Advent of new equipment and new methods could facilitate study on plant disease epidemiology. The new equipment and instruments contributed to modern epidemiology. Molecular tools, such as the DNA probes, allowed detection and identification of plant pathogens on plant surface, in a mixture of microbes and also in the vicinity of host plant. The PCR technique proved very useful for such purpose. Wrapped and scar markers are used in detecting genetic similarities. A computer system, the Geographic Information System, GIS, can be installed for assembling, storing, manipulating and displaying data to be referenced by geographic coordinates. GIS is often used for the spatial and temporal analysis of disease development over large geographic areas and helps determine the role and relative importance of various parts of these areas in the initiation and development of the epidemic. Global Positioning System, GPS, allows to pinpoint an individual tree or specific area or areas of the field affected by the pathogen. These areas can be examined at appropriate intervals. GPS can also be used to apply pesticides to areas where needed. GPS consists of a handheld device that is coordinated with a global system of satellites. Geostatistics is applied in epidemiology to characterize quantitatively spatial patterns of disease development or development of pathogen populations in space over time. Geostatistical techniques can also be used to study continuous and discrete variables. Remote sensing is measuring electromagnetic radiation reflected or emitted from an object. The instruments may be spectrum. Remote sensing instruments may be handheld, ground-based. 1951. Cameras with films and filters, digital cameras, video systems and radiometers, or they may be carried on balloons, aircraft, and satellites. Stored data are printed out and analyzed directly or after transferring to computer for creating their visual images. Image analysis. The photography and electronic analysis of large areas of fields or mountains also proved useful in epidemiological studies. Image or photographs are taken through aerial photography, ground-based sensor data, and satellite-borne and airborne sensors. Image analysis can provide a measure of the disease severity in each plant or area of infected plants. Information technology. The use of computers alone or in combination with other electronic devices is also very useful in epidemiology. They help collect data on diseases at various levels and locations. This technology can describe and display spatial patterns of characteristics of different pathogens, such as their genotypes at agricultural region levels. Disease, warning and expert systems. In many developed countries in Europe and USA, different types of warning systems are used for one or more important plant diseases. The purpose is to warn farmers of the impending onset of infection period or to inform them that an infection period has already occurred so that they can take immediate appropriate control measures to stop further infections. In most cases, the warning system begins with a grower, an extension agent, or a private consultant surveying certain fields on a regular basis. When mature inoculum, as ascaspores in apple scab, or traces of disease, as in potato late blight, are found, the county extension office is notified. This office in turn notifies the state extension plant pathologist, who collates all reports about the disease from around the state and by email, telephone, fax or in writing notifies all concerned county agents. Pest alert. They, in turn, notify all farmers in their county. For diseases of potential regional or national epidemic consequences, the state extension plant pathologist notifies the Federal Plant Disease Survey Office at the U.S. Department of Agriculture, which in turn notifies all extension plant pathologists in adjacent and other states that may be affected by plant disease. Since the mid-1970s, computerized warning systems are in use for certain diseases in some states. Some of them, such as Blickcast, are centrally located computers that process weather data either collected on the farm by individual growers and transmitted or phoned in when certain intervals. The computer has been processes the data, determines whether an infection period is imminent, likely to occur, or cannot occur, and makes a recommendation to the grower as to whether to spray and what materials to apply. After 1980, small, special-purpose computers have been developed that have field sensors and can be mounted on a post in farmer's field. Such units, such as the Apple Scab Predictor, monitor and collect data in the field on temperature, relative humidity, leaf wetness, and rainfall, analyze the data and make predictions of disease occurrence and intensity as well as recommendations on the spot for disease control measures. The same unit can be used for any disease for which a prediction program is available, in which case either the unit can be reprogrammed or the program circuit boards can be interchanged. 1952. Expert systems are computer programs that try to equal and, better yet, surpass the logic and ability of an expert professional in solving problems, the solutions of which require experience, knowledge, judgment and interactions. Expert systems can use data in almost any format, and can suggest solution to the problem. Expert systems in plant pathology are frequently used for diagnosis of a plant disease. Several expert systems also incorporate the decision-making process of the expert and advise growers in making decision on disease management in respect of kind, amount and time of application of pesticides, etc. In simplest form, the expert systems utilize a bank of data pertinent to the problem stored in the computer and also a knowledge base input by the expert, consisting of one or more, if conditions, followed by a conclusion or action, then action, and finally a recommendation. Usually, the expert system is also reviewed and if necessary, revised by other experts. In USA, Blickcast, 1975, which is computerized forecasting system for potato late blight, and the computer-based Apple Scab predictive system, 1980, are considered to be the processors to expert systems. The first expert system in plant pathology was developed in 1983 to diagnose nearly 20 soybean diseases in Illinois. Since then, expert systems have been developed for the diagnosis or management of diseases of tomato, grapes, wheat, concealer, peach and nectarine, calyx, apple, pomme, the Penn State Apple Orchard Consultant PASOK, wheat, more crop, etc. More crop. Managerial options for reasonable economical control of rusts and other pathogens is designed to provide disease management options in different geographic regions and agronomic zones of the Pacific Northwest. Some of the frames, windows, of the program show the wheat diseases about which one should be concerned. Brief information about each disease, suggestions for control through seed treatment and foliar spray, timing of spray, etc., and which diseases can or cannot be controlled through a particular treatment are shown in the relevant frames. In India, not much work has so far been done on disease forecasting. For some diseases, information on relationship between pathogen activity and temperature-moisture conditions have been worked out. There has been generated data for forecasting the blast disease of the Magneporte grisae on the basis of minimum night temperature range of 20-26 C coupled with a high relative humidity about 90% lasting for about 1 week during any of the 3 susceptible stages of host growth. The disease can be forecast. 1954. 4 Integrated Disease Management Principles of Plant Diseases Management. Information on etiology, symptoms, pathogenesis and epidemiology of plant diseases are intellectually interesting and scientifically justified, but most important of all, they are useful as they help in formulation of methods developed for successful management of disease and thereby increasing the quantity and improving the quality of plant and plant products. Practices of disease management vary considerably from one disease to another depending upon the type of pathogen, the host and the biotic and abiotic factors involved. Methods for plant diseases control were first classified by Wetzel, 1929, into exclusion, eradication, protection and immunization. Further advances in plant pathology leading to development of newer methods. 2 more principles, avoidance and therapy, were created. 1. Avoidance. It involves avoiding disease by planting at time when, or in areas where inoculums is absent or ineffective due to environmental conditions. The major aim is to enable the host to avoid contact with the pathogen or to ensure that the susceptible stage of the plant does not coincide with favorable conditions for the pathogen. The main practices under avoidance are choice of geographical area, selection of the field, choice of sowing planting time, selection of seed and planting material, short-duration disease-escaping varieties and modification of agronomic cultural practices. For example, bean anthracnose is common in wet areas. Similarly, smut and ergot of pearl millet are serious in areas where rainfall occurs for long durations during flowering of the crop. The potato cultivation at high altitude is relatively free from viruses, as prevailing environmental conditions do not permit the buildup of vector populations. Similarly, early planting of potato or wheat in Indo-Gangetic plains may escape late blight or stem rust damage respectively. Exclusion. It means preventing the inoculums from entering or establishing in a field or area where it does not exist. Seed certification, crop inspection, eradication of inoculums and or insect vectors, and quarantine measures are some of the means of preventing the spread for pathogens. For example, the outbreak of citrus canker, Xanthomonas country's Axonopodis, due to heavy in USA destruction, in 1910 through strict planting material imported from Southeast Asian quarantine was imposed against entry of citrus planting material. In India, interstate quarantine is of in potato place for what the Syncytium movement of Endobioticum, potato from Darjeeling which is restricted area of West Bengal to prevent the spread to that area only. 1955. 3. Eradication. The process of reducing, inactivating, eliminating or destroying inoculums at the source, either from a region or from an individual plant in which it is already established is termed as eradication. Eradication involves eliminating the pathogen from infested areas. The magnitude of the operation involved may vary considerably. One of the most extensive eradication operations carried out so far was to get rid of the citrus canker, Xanthomonas axonopodis, in the USA during 1927 to 1935. As many as 4 million citrus trees were cut and burned at a cost of about $2.5 million to eradicate the pathogen. The practices invariably employed to achieve eradication of inoculums include eradication of alternate and or collateral hosts, crop rotations, field sanitations, heat or chemical treatments of plant materials or soil, biological control, etc. Spraying or dusting with sulfur to kill the mildew mycelium. 4. Protection. The protection of infection coats against the inoculums of many fast-spreading infectious pathogen, brought by wind from neighboring fields or any other distant place of survival. Principles of avoidance, exclusion and eradication may not be sufficient to prevent the contact of host with pathogen, thus development of the disease is imminent. Measures are necessary to protect host plants from invading inoculums. It can be achieved by creating toxic barrier between the plant surface and the inoculums. Methods employed to achieve such results are chemical sprays, dusts, modification of environment, and modification of host nutrition. Fungicidal sprays that act as protectants are used to control Cercospora leaf spot of sugar beet, especially in those fields where inoculum has carried over from the previous year. Sulfur is used as a protectant fungicide to control powdery mildew of sugar beet. 5. Host resistance. It utilizes in-built mechanism to resist various activities of pathogen. The infection or subsequent damage by pathogen can be rendered ineffective through genetic manipulation or by chemotherapy. The host resistance can also be induced by use of certain biotic and abiotic factors. The discovery of Mendelian laws of inheritance and developments in plant breeding techniques have helped in developing crop varieties resistant to specific pathogen or group PF pathogens. The classical breeding techniques include selection, mutation and hybridization. Use of biotechnological tools such as tissue culture, genetic engineering and protoplast fusion are being used to develop resistant cultivars of various economically important crops. 1956. 6. Therapy. It is the treatment of infected host plant, which is attempted in case of economically important horticulture plants. As a principle of plant disease control, it provides an opportunity to cure or rejuvenate the diseased host plant by use of physical or chemical agents. Chemotherapy is the use of chemicals to inactivate the pathogen, whereas heat is sometimes used to inactivate or inhibit virus development in infected plant tissues so that newly developing tissue may be obtained which is free of pathogen. Thermotherapy involves the exposure of diseased plants or parts of them to hot water or high air temperature for different periods of time. Loose smut of wheat is controlled by treating the seeds with hot water, but growing resistant varieties is a simpler method of control. Hot water treatment has been used to kill nematodes in bulbs, combs, tubers and fleshy roots while they are in a dormant condition. The first 5 of these principles are mainly preventive, prophylactic, and constitute the major components of plant disease management. They are applied to the population of plants before infection takes place. Therapy is a curative procedure and is applied to individuals after infection has taken place. Under the concept of disease management, these principles have been classified into following 5 categories: management of physical environment, cultural control, management of associated microbiota, biological antagonism, management of host genes, host resistance, management with chemicals, chemical control, management with therapy, physical, chemical, etc. Physical methods. The scientific principle involved in heat therapy is that the pathogen present in seed material is selectively inactivated or eliminated at temperatures that are non-lethal to the host tissues. Following physical methods are employed for reduction or elimination of primary inoculums that may be present in seed, soil or planting material. The physical agents used most commonly in controlling plant diseases are I. Temperature, high or low. 2. Dry air. 3. Unfavorable light wavelengths. 4. Various types of radiations. V. Cultivation in glass or plastic greenhouses. 6. Plastic or net covering. 1956. Hot water treatment, HWT. The seeds are soaked in cold water at 20-30 C for 5 HRS to induce thidomant mycelium to grow. Then the seeds are immersed in hot water at 50-54 C for 10 minutes to kill demycelium. It is very effectively used to eliminate loose smut of wheat. The sets of sugarcane can be treated at 50 C for 2 HRS to eliminate grassy shoot pathogen. The main drawback in the hot water treatment is that the seeds may be killed or lose its germinability if the period of treatment exceeds the specified time. So this method is replaced by other physical methods like hot air and aerated steam treatment, wherein the seeds are exposed only to hot air, aerated steam. Hot air treatment, HAT, sugarcane sets are treated with hot air at 50 C for 2 HRS to eliminate mosaic virus. Aerated steam therapy, AST, sugarcane sets are also exposed to aerated steam at 50 C for 3 HRS to eliminate mosaic virus. Moist hot air treatment, MHAT, this method is effectively used in sugarcane to eliminate grassy shoot disease. Initially the sets are exposed to hot air at 54 C for 8 HRS, then exposed to aerated steam at 50 C for 1 HR, and finally to moist hot air at 54 C for 2 hours. Solar heat treatment, SHT. A simplest treatment has been devised in India to eliminate the pathogen of loose smut of wheat. Previously the hot water treatment was followed to eliminate loose smut. As the terminal death point of the fungus and the embryo are very close, the extensive care should be taken to avoid killing of the embryo. Luthra in 1953 devised a method to eliminate the deep-seated infection of Ustilago nuda. The method is popularly known as solar heat or solar energy treatment. Luthra's solar energy treatment, the seeds are soaked in cold water for 4 hours in the forenoon on a bright summer day, followed by spreading and drying the seeds in hot sun for 4 hours in the afternoon. Then, the seeds are again treated with carboxin or carbendazim at 2 g/kg and stored. This method is highly useful for treating large quantities of the seed lots. 6. Soil solarization. Soil solarization is generally used for controlling soil-borne pathogens like Pythium, Verticillium, Rhizoctonia, Fusarium etc. and nematodes in small areas like nurseries. Irrigate the nursery bed to moisten the soil to a depth of 10 centimeters. Cover the bed after 2 days with thin transparent polyethylene sheets for 4 to 6 weeks and then irrigate the beds once in a week. The purpose of irrigation is to increase the thermal sensitivity of resting structures of fungi and to improve heat conduction. Steam sterilization. Soil sterilization is completed when the temperature in the coldest part of the soil has remained for at least 30 minutes at 82 C or above, at which temperature almost all plant pathogens in the soil are killed. 1957. The soil is steam sterilized either in special containers, soil sterilizers, into which steam is supplied under pressure, or on the greenhouse benches, in which case steam is piped into and is allowed to diffuse through the soil. Steam is passed through perforated pipes at a depth of 15 centimeters to sterilize the upper layers of soil. It is mostly practiced under glasshouse and greenhouse conditions. At about 50 C, nematodes, some oomycetes, and other water molds are killed, whereas most plant pathogenic fungi and bacteria along with some worms, slugs, centipedes, are usually killed at temperatures between 60 and 72 C. Most weeds, rest pathogenic bacteria, most plant viruses in plant debris, and most insects are killed at about 82 C. Heat-tolerant weed seeds and some plant viruses, such as tobacco mosaic virus, TMV, are killed at or near the boiling point that is between 95 and 100 C. Hot air sterilization. Hot air is also passed through pipelines to sterilize the soils in the nursery areas. 9. Hot water treatment. It is mainly done in pot culture studies to kill the fungi and nematodes. The pots containing soil are immersed in boiling water at 980 C for 5 minutes or drenching boiling water at 20 liters SQ.m. Refrigeration. It is an accepted fact that the low temperature at or slightly above the freezing point checks the growth and activities of all such pathogens that cause a variety of post-harvest diseases of vegetables and fruits. Therefore, most perishable fruits and vegetables should be transported and stored in refrigerated vehicles and stores. Cool chains refrigerated space from field to consumer table is becoming very popular. Regular refrigeration is sometimes preceded by a quick hydro-cooling or air cooling to remove the excess heat carried in them from the field to prevent development of new or latent infections. Disease control by irradiation. In this method, various electromagnetic radiations are used for controlling post-harvest diseases of fruits and vegetables by killing the pathogens present on them, such as UV light, X-rays, gamma rays, particulate radiations, such as A particles and particles. It has been possible to control diseases on greenhouse vegetables caused by species of these fungi by covering or constructing the greenhouse with a special UV-absorbing vinyl film that blocks transmission of light wavelengths below 390 nm. 1957. 2 regulatory methods or legislative methods, plant quarantine and inspection. Quarantine can be defined as a legal restriction on the movement of agricultural commodities for the purpose of exclusion, prevention or delay in the spread of plant pests and diseases in uninfected areas. Quarantine measures are of 3 types. I. Domestic. 2. Internal. 3. Total embargoes. The quarantine law was first enacted in USA in 1912 and was known as Federal Quarantine Act. In India, the Destructive Insect and Pest Act, DIPA, was passed in 1914 and subsequently supplemented by other provisions. Such quarantine laws were first enacted in France in 1660, and in Denmark in 1903. They aimed at the rapid destruction or eradication of barberry, which has been known since early times to harbor black rust pathogen. Diseases believed to have been introduced into India from foreign countries. Disease host date of first record introduction from leaf rust, Hemilia vasterix, coffee 1879, Sri Lanka late blight, Phytophthora infestans. Potato 1883 Europe downy mildew, Plasmodium viticola, grapevine 1910 Europe downy mildew, Pseudoperonospora cubensis, cucurbits. 1910 Sri Lanka plant quarantine methods. There are number of plant quarantine methods which are used separately or collectively to prevent or retard the introduction and establishment of exotic pests and pathogens. The components of plant quarantine activities are 1. Complete embargoes. It involves absolute prohibition or exclusion of specified plants and plant products from a country infected or infested with highly destructive pests or diseases that could be transmitted by 1958. The plant or plant products under consideration and against which no effective plant quarantine treatment can be applied or is not available for application. 2. Partial embargoes. Partial embargoes, applying when a pest or disease of quarantine importance to an importing country is known to occur only in well-defined area of the exporting country and an effectively operating internal plant quarantine service exists that is able to contain the pest or disease within this area. 3. Inspection and treatment at point of origin. It involves the inspection and treatment of a given commodity when it originates from a country where pest disease of quarantine importance to importing country is known to occur. 4. Inspection and certification at point of origin. It involves pre-shipment inspection by the importing country in cooperation with exporting country and certification in accordance with quarantine requirements of importing country. 5. Inspection at the point of entry. It involves inspection of plant material immediately upon arrival at the prescribed port of entry and if necessary subject to treatment before the same related. 6. Utilization of post-entry plant quarantine facilities. It involves growing of introduced plant propagating material under isolated or confined conditions. Agencies involved in plant quarantine. The authority to implement the quarantine rules and regulations framed under DIP Act rests basically with the Directorate of Plant Protection, Quarantine and Storage, under the Ministry of Agriculture. This organization handles bulk import and export of seed and planting material for commercial purpose. Under this organization 9 seaports, 10 airports and 7 land frontiers are functioning. These are the recognized ports for entries for import of plant and plant material. The Government of India has also approved 3 other national institutions to act as official quarantine agencies, especially for research material. 1. National Bureau of Plant Genetic Resources, NBPGR. The NBPGR in New Delhi and its regional station at Hyderabad in the agency involved in processing of germplasm, seed, plant material of agricultural, horticultural, and silvicultural crops of all the institutions of Indian Council of Agricultural Research, ICAR, functioning in the country. It is also responsible for quarantine clearance of seed and plant material received from international agricultural research centers with ICRISAT, ICARDA, CIMIT, etc. ICRISAT was established in 1972 at Patancheru, near Hyderabad, to work on improvement of sorghum, pearl millet, chickpea, pigeon pea and groundnut. The quarantine clearance of all its exchanges was handled by Centra, Plant Protection Training Institute of 2152. Directorate of Plant Protection, Quarantine and Storage, until July 1986. This authority was later passed on to NBPGR in August 1986. Forest Research Institute, Friday, Dehradun, for forestry plants and Botanical Survey of India, BSI, for other plants. The various steps involved in import quarantine clearance of seed and propagating plant material is outlined below. Securitization of import application filed along with attached documents such as phytosanitary certificate, original, permit, importer's copy, shipping bill, invoice, packing list and customs bill of entry etc. to ensure the import is in order and that no prohibited plant material is imported. Assessment of inspection fees and registration of application. Inspection and sampling of the consignment at port warehouse or container terminal. Sampling of seed usually carried out as per the provisions of ISTA rules and regulations. Whereas in case of bulk import of vegetative planting materials such as cuttings, saplings, budwoods, bulbs, tubers, etc., at least a minimum of 0.1% of propagules are sampled variety and examined to ensure free from exotic pests or pathogens. In case of quarantine pests suspected, 100% inspection is carried out for critical assessment of the risk. 4. Detailed laboratory testing. Visual inspection. The samples of seed propagating plant material is examined with the help of illuminated magnifier to record live insect infestation, contamination by soil and weed seeds, nematode galls, sclerotia, smut bunt balls, etc. Sometimes inspections are carried out under UV lamp to facilitate detection of specific seed-borne inspection by characteristic fluorescence. X-ray test for detecting hidden insect infestations such as bruchids and weevils that bore into seed. Washing test to detect surface-borne oospores of downy mildew, smut spores, bunt spores, etc. and nematode cysts. Seed samples of onion, clover and lucerne are soaked for 24 to detect stem and bulb nematode and also root washings are examined for ectoparasitic nematodes. De-incubation tests such as blotted test or agar plate test carried out for detecting seed-borne pathogens such as fungi. Fluorescent Pseudomonas agar used for selective detection of seed-borne bacteria. E-grout test coupled with indicator inoculation tests for detecting seed-borne viruses and bacteria. Besides this, special diagnostic tests such as electron microscopy, DIP method, enzyme-linked immunosorbent assay, ELISA, are used for detection of specific viruses in the imported seed planting material. Penicillinous-based DAC ELISA is widely used for the detection of virus in imported seed plant material. The detailed testing procedures for the detection of seed-borne pathogens are outlined in the seed health testing chapter. Fumigation and treatment techniques. Fumigation is the versatile technique used for eliminating insect infestation. Methyl bromide is the most commonly employed for controlling insect infestation and readily adopted in 2153. Quarantine programs as the exposure time involved is short and affect all stages of insect pests and high penetrating power. 2 types of fumigation with I. Atmospheric fumigation under gas-proof sheets or chambers and 2. Vacuum fumigation in vacuum chamber is widely employed. The other chemical treatments include insecticidal fungicidal drippings or spraying or seed dressings are invariably associated with growing under post-entry quarantine conditions. The temperature treatments such as hot water treatment, hot air treatment or vapor heat treatment are carried out to control internally borne infection infestation and the latter particularly employed to control fruit fly infestation. Cold treatments such as refrigeration to control insect infestation in fresh fruits and vegetables. Of late, irradiation is used to control insect infestation and spoilage of food products during storage and as well as application of high-intensity electronic beams through an accelerator is under experimentation. Certification. Phytosanitary or health certificate is a certificate which should accompany a plant or plant material or seed which is to be moved from one place to another place. This certificate indicates or certifies that the material under transit is free from pests or diseases. A model phytosanitary certificate proposed at the government consultation on the International Plant Protection Convention at Rome in 1976 and approved by FAO in 1979. Domestic quarantine. Under the DIP Act, the Directorate of Plant Protection, Quarantine and Storage has the responsibility to take the necessary steps and regulate the interstate movement of plants and plant material in order to prevent the further spread of destructive insects and diseases that have already entered the country. The sole object of enforcing domestic quarantine is to prevent the spread of these diseases from infected to non-infected areas. Currently, domestic plant quarantine exists in 4 diseases: wart, Syncytium endobioticum, of potato from 1959, bunchy top, virus, of banana from 1959, mosaic, virus, of banana from 1961 and apple scab, Venturi inequalis, from 1979. Most of the states in India have plant quarantine laws to avoid entry of plant pests and diseases. Bunchy top of banana, the export and the transport from the states of Assam, Kerala, Orissa, West Bengal, Tamil Nadu to any other state of banana plant or any other plant of the genus Musa, including sucker, stem, leaf, flower, and any other part thereof which may be used for propagation, or the materials of banana plant or any other plant of the genus Musa, which are used for packing and wrapping, excluding the banana fruit is prohibited. Banana mosaic, the export and transport from the states of Maharashtra and Gujarat of any plant of banana or any other plant of genus Musa including the sucker, stem, flower and any other part thereof, but excluding leaf and fruit thereof is prohibited. 2153. Potato wart, the export to potato tubers from the state of West Bengal to any other state or territory of India is prohibited. Apple scab, the Directorate of Horticulture, Himachal Pradesh worked out a detailed scheme for the eradication of scab. Limitations. There are many limitations to implementing domestic plant quarantine in India due to the vastness of the country and the unrestricted movement of plant material from one state to another. As a result, the diseases like bunchy top and mosaic of banana have spread to several other states. However, the wart disease, golden nematode of potato, and scab of apple are restricted in the states where they were initially noticed. In India, the Directorate of Plant Protection, Quarantine and Storage, DPPQS, under the Ministry of Agriculture implements the plant quarantine regulations for bulk consignments, and National Bureau of Plant Genetic Resources under Indian Council of Agricultural Research, ICAR, is the nodal agency for safe movement of germplasm including transgenics. This work under the Plant Quarantine Regulation of Import in India Order 2003 which came into force from 1 January 2004, under the Destructive Insect and Pests Act of 1914. Although the regulations are now in place, there are a number of issues related to quarantine of germplasm as it has been drafted more for facilitating bulk imports than for exchange of germplasm. Under this order a pest risk analysis, PRA, has been made mandatory and the various schedule of the order give lists of crops for which a generic PRA is given. A number of cultivated crops, and their germplasm including wild relatives land races, do not find mention in any of the schemes. Hence, a detailed PRA becomes obligatory for them prior to import. The size of sample consignment, technique to be used for detection of minute amounts of pest, availability of diagnostic reagents and reference collection of exotic pests, and post-entry quarantine testing are important technical issues in quarantine. Over the years, during quarantine processing, the pests intercepted include many like late blight of potato, banana bunchy top, bacterial blight and streak disease of paddy etc. that are not known to occur in India, have different races biotypes strains not known to occur in India, are present on new host, are from a country from where they were never reported before or are an entirely new pest species hitherto unreported in science or are reported to be present widely in India. These interceptions, especially of pests not yet reported from India signify the importance of quarantine in preventing attack of destructive exotic pests. The need of the hour is to strengthen the agricultural biosecurity system of the country. Cultural methods. Eradication. Eradication is the elimination of pathogen after it has become established in the area where host is growing. The following are the important methods followed to prevent the spread of the disease. I. Eradication of alternate hosts. 2154. Il. Eradication of collateral and self-sown overwintering hosts. 3. Eradication of the affected plants or trees. 4. Eradication of pathogens from infected plant parts by surgery and Eradication of culled out plant materials, debris, etc., through different cultural practices. Eradication of alternate hosts. Removal of alternate hosts helps to prevent and check the spread of the disease caused by heterococcus rust pathogens in the primary hosts. Barberry bush is the alternate host for black stem rust pathogen Puccinia graminis tritici on wheat, where the pathogen survives in the off-season. Barberry was eradicated in Canada, Denmark, France, Hungary, Norway and in the USA by passing stringent laws in each country. The eradication of barberry had two benefits: it elimination of early spring primary inoculum and prevention of the formation of new physiologic races of the pathogens. Eradication of collateral and self-sown overwintering hosts. There are many weed hosts or wild species of cultivated plants act as collateral hosts or volunteer plants of an economic crop which act as reservoirs of pathogens of annual crop. Reservoir hosts help the pathogen to continue the infection chain. The primary inoculum is produced on and dispersed from these hosts to the cultivated crop hosts. If these wild or uneconomic host plants of the pathogen are destroyed, the sources of primary inoculum are eliminated and chances of initiation of the disease in the crop hosts are reduced. Destruction of these hosts breaks the life cycle of the pathogen and the infection chain. Reservoir hosts or indigenous plant species which are not actually involved with the life cycle of the pathogen but provide additional sites for its persistence and multiplication. In some cases, such plant species act as symptomless carriers, especially for viruses and root pathogens. 2155. Crop disease pathogen collateral hosts. A. Fungi blast Pyracularia oryzae Brachyria mutica. Rice panicum ripens sorghum ergots Phacelia sorgi panicum spp. B. Bacteria bacterial leaf Xanthomonas oryzae pv. Cyanodon dactylon, rice blight oryzae Cyperus rotundus, apple and pear fire blight Irvinia amylovora hawthorn bushes Crataegus sp. Cotton bacterial blight X. Axonopodis jatropha kirkas pv. Malvacearum C. viruses yellow mosaic virus sweet clover 1. Bean. 2159. Phytoplasma brinjal little leaf Phytoplasma catharanthus roseus datura sp. Crop rotation. Crop rotation is essentially a preventive measure and has its effect mainly on the succeeding crop. Crop rotation is the oldest and cheapest method adopted in agriculture for eradication of certain types of pathogens from infested soil. Continuous cropping or monoculturing provides opportunity for perpetuation of pathogenic organisms in the soil when the same crop is raised year after year in the same field. The soil-borne pathogens of that crop easily perennate in the soil and increase in their population. After some time, the soil becomes so heavily infested that it becomes unfit for cultivation of the particular crop. Virus diseases of crop plants and their vectors are found to increase after every crop if a crop is cultivated continuously in a field. On the other hand, when immune, resistant or non-host crops are grown for a definite duration after a susceptible crop in the field, it is expected that in the absence of nutrition, the pathogen will be starved of and the population of such pathogens consequently decreases. It is also possible that different crops release some biochemical substances in their root exudates which either directly kill the pathogen or encourage development of antagonistic microorganisms in the soil. In this way, crop rotation is one of the most effective methods of root disease control. Organisms which are soil inhabitant types remain in soil for a very long time, even more than 5 years in the absence of the host. Long-lived spores or the organisms by themselves subsist as saprophytes and therefore their presence in soil is long-term. Onion smut, Urocystis sciepulae, and clubroot, Plasmodiophora brassicaceae, organisms are producing resistant type of spores while Rhizoctonia, Fusarium and some species of Pythium are those which could remain in soil as saprophytes for a very long time. Eradication of such organisms becomes fairly difficult. Soil also harbors soil invaders. These organisms are not persistent and they can live as long as the host residues serve as substrate. They perish when they are forced to exist in the soil in competition with soil inhabitants and disappear gradually in due course. Bean anthracnose fungus Caletotrichum lindemutinum, cabbage black rot bacterium, Xanthomonas campestris pv. Campestris are some examples, which live in soil for 1 to 2 years. They can be eliminated from soil by adopting 3 or 4 year rotation with non-host crops. Crop rotation is effective in the control of brown stem rot of soybean, Cephalosporium grugatum. The disease incidence can be reduced to a great extent by rotating with corn for 4 to 5 years between 2 soybean crops. Crop rotation with sugarcane or paddy is effective in the control of Panama wilt, of banana, Fusarium oxysporum f.sp cubans, and crop rotation with paddy or green manures is effective in the control of red rot of sugarcane, Caletotrichum falcatum. Rotation of cereal crops like pearl millet, finger millet or foxtail millet is recommended for the control of Macrophomina root rot. 22 o'clock. Of pulse crops. 2-year crop rotation with lucerne is recommended in the control of verticillium wilt of cotton. Many diseases such as Fusarium wilt of pigeon pea, F. udum, foot rot of betel vine, Phytophthora capsicum, bacterial leaf blight of rice, Xanthomonas oryzae pv oryzae, bacterial blight of cotton, X. campestris pv malvacearum, etc., are controlled by this method. 3. Fallowing. Fallowing starves the pathogen and helps in reduction of the inoculum by elimination of the host. Diseases like Macrophomina root rot on different crop plants is controlled by following this method. Flood fallowing is to a depth of 0.6 to 1.5 m for 4 to 6 months markedly reduced the Panama wilt pathogen Fusarium oxysporum f.sp. cubans inoculum in banana. Wet fallowing reduces saprophytic survival of Alternaria solni on crop debris. 4. Application of organic manures. Addition of organic manures like farmyard manure or green manures or oil cakes to the soil increases the antagonistic microorganisms in the soil. Build-up of antagonistic microorganisms reduces the population of soil-borne plant pathogens and the diseases caused by them. Application of farmyard manure at the rate of 12.5 tons ha reduced the incidence of macrophomina root rot of cotton. Application of 5 kilograms of neem cake tree 3 times in a year reduces the basal stem rot, Ganoderma lucidum, of coconut. 5. Summer plowing. Deep plowing during summer periods buries the inocula of fungi of soil-borne nature. Fungal propagules, sclerotia and different types of spores conidia on plant refuses die when exposed to sunlight due to the higher temperature prevailing during the summer. Further infected self-sown plants, volunteer hosts plants, weed hosts, regrowths from the plant roots, alternate hosts and alternative hosts are also destroyed. Here, the spread of the disease is avoided. Groundnut blight, Cotischem rolfsill, is controlled by plowing the soil to a depth of 20 centimeters. The inverted plow soil. Soil buries the sclerotia of the fungi, claviceps, sclerotium and sclerotinia in association with plant or alone, impedes the discharge of ascospores from perithecia. Bunt and smut spores of wheat, smut spores of sugarcane and sorghum and microsclerotia of verticillium in cotton are buried deep into the soil by deep plowing. 6. Crop growing seasons. Rice blast becomes serious when the rice crop is raised from August to September in Tamil Nadu. Ragi blast becomes serious when sowing is made between June and August. Similarly, yellow mosaic of black gram, green gram and phyllodes of sesame are serious during Kharif season in South India. Incidence of powdery mildews of different crops is found to be high during Rabi when compared to Kharif and summer seasons. In Bhendi, yellow vein mosaic incidence is very high during summer. The seasons with high incidence of diseases should be avoided in the epidemic areas. 2206. 7. Selection of seeds and seed materials. Seeds and seed materials carry many fungi, bacteria, viruses and phytoplasmas and may introduce these pathogens into the field. I.e. Seeds and seed materials form the primary source of infection. Seed and seed materials like cuttings, tubers, grafts, sets etc. should be well matured, disease-free, uninjured and have a high germinating capacity. The absence of an initial inoculum in seeds is definitely helpful in delaying or suppressing incidence of the disease. It is a preventive method. Diseases like foot rot, brown spot short smut of sorghum, loose smut of wheat, bacterial blight of rice, bacterial blight of cotton, leaf crinkle of black gram etc. are transmitted through seeds. Virus diseases and black ring of potatoes, foot 8 rot of ginger, foot rot of betel vine, Panama disease of banana, red rot of sugarcane cassava mosaic, bunchy top and virus diseases of fruit trees are transmitted through tubers, sets, rhizomes, cones, grafts and budwoods. Tuber indexing is a special method to obtain disease-free seed materials in potato. It is commonly practiced by nurseries and seed merchants selling potato seed tubers. Use of seeds in the place of rhizome sucker is recommended in the control of cuttay, disease of cardamom. 8. Burning of stubbles and crop residues. Burning of plant wastes, crop residues, stubbles, etc. in the areas selected for raising nurseries for vegetable crops, tobacco, chilies and forest trees etc. heats the soil and kills the inoculum of the pathogens present in the top layer of the soil. When nurseries are raised in these areas, incidence of damping of disease is highly reduced. This practice is also followed in pits made for planting coconut, banana, fruit trees etc. Burning of rice crop residues avoid carryover of sheath blight, Rhizoctonia solni, stem rot, Sclerotium oryzae, of rice and bacterial blight of cotton. 9. High budding. High budding is a practice to avoid infection by gummosis fungus of citrus trees. In low-budded plants, the bud point is close proximity to infection center, the soil, become readily diseased. High budding is a simple device for lengthening this distance between the bud point and infected soil. In this method, the soil-borne pathogens, Phytophthora palmivora and P. sacrophthora, have no chance of reaching the bud point, through which they enter the bark. Staking of lower-most branches arising close to the soil increases the distance between the fruits and soil inoculum and removes the chances of brown rot, Phytophthora sp. infection and buckeye rot of tomato, P. necotianae var. Parasitica. 10. Intercropping. Intercropping is also a device in the control of some soil-borne diseases. Intercrops should be properly chosen so that they should not have any common pathogen. For E.g. Macrophomina phaseolina has got wide host range and hence common host should not be grown as intercrops. 2208. Intercropping with moth bean, Phaseolus aconitifolius, in a cotton field reduced the root rot, M. phaseolina, incidence. Due to reduction in the number of host plants, there is sufficient spacing between them and chances of contact between foliage of roots of diseased and healthy plant are greatly reduced. Therefore, root pathogens are unable to spread from diseased to healthy roots and spread of foliar pathogens is also reduced to a great extent. Intercropping of sorghum in pigeon pea field reduced the wilt, F. udum, incidence. The roots of non-host plants may act as a barrier obstructing the movement of pathogens in soil. They may release toxic substances from their roots which may suppress the growth of the pathogens attacking the main crop. GAU. Hydrocyanic acid, HCN, in root exudates of sorghum is toxic to F. udum, the pigeon pea wilt fungus. Intercropping of sorghum of moth bean in a crop of cluster bean reduced the incidence of root rot, R. solni, and wilt, F. coruleum, from 50 to 60% in single crop to 8 to 15% in the mixed crop. Barrier cropping. Taller crops can be grown to protect a crop of lesser height from virus vectors. The insects may land at the taller crops, barrier crops, and the dwarf crop may escape from virus diseases by those insects. Barrier cropping with 3 rows of mudge or sorghum or pearl millet around the main crop, namely black gram or green gram, is effective in reducing the vector population and incidence of yellow mosaic. Another best example is growing of 3 rows of kale or barley as barrier crops in cauliflower seed beds and undersown beets tackling against cauliflower mosaic and beet yellows diseases respectively. The incoming aphids are thought to land on the barley or kale and probe briefly, causing them to lose the non-persistently transmitted virus they are carrying. Maize or sunflower are the other barrier crops considered for these crops. Decoy crop and trap crop. Decoy crops, hostile crops, are non-host crops sown with the purpose of making soil-borne pathogens waste their infection potential. This is effected by activating dormant propagules of fungi, seeds of parasitic plants, etc. in absence of the host. A list of pathogens that can be decoyed is given in table 2211. Trap crops are host crops of the pathogen, sown to attract pathogens but destined to be harvested or destroyed before they complete their life cycle. Fodder sorghum can be raised as a trunk crop to reduce downy mildew of sorghum. 13 Yellow sticky traps. Sticky, yellow polythene sheets erected vertically on the windward side of red pepper fields have been shown to reduce the incidence of potato virus Y, PVY, and cucumber mosaic virus, CMV, in the crop. The aphids are attracted to the yellow color and are caught on the sticky polythene. The control obtained was so successful that the method has become a standard control procedure in red pepper crops in Israel. Similar traps have also been used to protect seed potato crops against potato leaf roll virus. Yellow sticky traps are in use to attract and kill the whitefly vectors which spread yellow mosaic of black gram and green gram and drain the yellow vein mosaic. Irrigation water management. Irrigation to the crop in the field is to wet the soil to the extent that roots easily get water and nutrients. If excess water is added to soil, it may directly affect activity of pathogens and or it may affect disease incidence through the effect on the host. Scab attack on potato tubers is prevented by maintaining soil moisture near field capacity during tuber formation. Bacterial flora antagonistic to Streptomyces scabies increases under high moisture conditions. The charcoal rot pathogen, Macrophomina phaseolina, attacks potatoes and cotton when the soil temperature rises and there is water stress. By irrigating the field, soil temperature is brought down, stress is removed and the disease is suppressed. When excess irrigation is made, the juvenile stage of plants is lengthened, making it susceptible to attack of fungi like Pythium. Supply of frequent but low quantity of irrigation water is, therefore, recommended for reducing chances of damping of in nurseries. Field and plant sanitation. Field and plant sanitation is an important method of disease control through cultural practices. The inoculum present on field plants in the field may multiply on the plant or in the soil and in due course of time may be sufficient to nullify or reduce the effect of control practices. Many pathogens overwinter or oversummer on plant debris during the off-seasons and become active when the crop is again grown in the field. Hence plants bearing pathogens or plant debris introducing inoculum into the soil should be removed as early as possible. In most of the soil-borne diseases like wilt and root rot, it has been reported that as long as the dead roots and other affected parts are present in the soil, the fungus continue its growth. When such diseased plant materials are removed, there is quick decline in the population of pathogens in the soil. In this manner, fusarium wilt of cotton, pigeon pea and banana, verticillium wilt of cotton, root rot of beans, downy mildew of pearl millet, sorghum, maize and peas, foot rot of betel vine, bacterial blight of cotton, white rust of crucifers, black spot of rose, powdery mildew of pea and cereals are reduced. Management of plant nutrients. The plant nutrients in general when applied in excess may increase or reduce the resistance in plants to diseases. Increased application of nitrogenous fertilizers increases the incidence of many 2212 diseases. Crops fed with heavy doses of nitrogenous fertilizers grow robust with foliage and succulent tissue but become highly susceptible to the attack of diseases like rust, powdery mildew, blast, tobacco mosaic and some bacterial diseases. In the case of blast of rice, optimum dose of nitrogenous fertilizers are recommended and it is applied in 3 split doses with 50% as based at transplanting, 25% at tillering and 25% at panicle initiation stage. Late application of nitrogenous fertilizers increases wheat leaf blotch, Septoria nodorum, and powdery mildew, Erysiphe graminis triticea. Some diseases are favored by ammoniacal form of nitrogen while others are favored by nitrate form of nitrogen. In general, wilts, fusarium sp, and root rots, Rhizoctonia spp, are favored by ammoniacal nitrogen while verticillium wilts and root rots due to Pythium spp are favored by nitrate nitrogen. In rice, blast disease is favored by ammoniacal nitrogen while brown spot, Helminthosporium oryzae, is favored by nitrate nitrogen. 4. Biological control. Biological control is defined as the reduction of inoculum density or disease-producing activities of a pathogen or parasite in its active or dormant stage by one or more organisms accomplished naturally or through manipulation of the environment, host or by introduction of one or more antagonists or by mass introduction of one or more antagonists. Biological control is but control of plant diseases using living microorganisms. Root rot disease, Macrophomina phaseolina, is a major disease in pulses, oilseeds, cotton, etc., and the most common method of control is using fungicides. But the chemical methods are uneconomical and low effective, as seed treatment with chemical may give protection only in the early stages of crop growth 2 weeks. In addition, it is harmful to the beneficial microorganisms in soil and creates residual problems. So, the biological control can be very efficacy used for the root rot disease management as the biological agent multiply in soil and offer protection throughout the crop growth. The 4 main mechanisms involved in the biocontrol are I. The biological agent, antagonist, may parasite the other organism, 2. Antagonist may secrete metabolites, antibiotics, harmful to the pathogens, antibiosis. 3. Antagonist may compete with the pathogens for nutrients or space, competition. And 4. May cause death of the parasite by producing enzymes, lysis. Parasitism and lysis. The biocontrol against parasitases the pathogen by coiling around the hyphae, E.g. Trichoderma viride. Various bacteria and fungi secrete hydrolytic about the degradation of cell wall of pathogens, e.g. I. Bacillus sp. causes hyphal lysis of Geomenorrhaeus graminis. 2. The chitnolytic enzymes of Serratia marcescens caused cell wall lysis of Cyrotium rolfsi. 3. Trichoderma sp. produces chitinases and 1,3-glucanases which lyses the cell wall of Rhizoctonia solni. Antibiosis. The antibiotic compounds secreted by the biocontrol agent suppress the growth of the pathogen, e.g. phenazine-carboxylic acid produced by Pseudomonas fluorescens plays an important role in suppressing the take-all disease of wheat. 2213. Competition. The biocontrol bacteria and fungi compete for food and essential elements with the pathogen thereby displacing and suppressing the growth of pathogen, e.g. I. The competition for nutrients between Pythium aphanidomachum, P. ultimum and bacteria suppress the damping of disease in cucumbers. 2. Fluorescent siderophores, iron chelators, such as pseudobactenes and puverdins produced by P. fluorescens chelates iron available in the soil, thereby depriving the pathogen of its fair requirements. The fungus, Trichoderma viride is one such biocontrol agent, mainly used for the control of root rot diseases of pulses and oilseeds. Trichoderma herzianum, for the control of several plant pathogenic fungi. Bacillus subtilis. This bacterium is widely used for the control of soil-borne plant pathogens like Macrophomina phaseolina, Rhizoctonia solni, Fusarium spp etc. This treatment also considerably improves the plant growth and yield. Bacillus subtilis is a rod-shaped, thermophilic gram-positive, aerobic bacterium. Pseudomonas fluorescens. This is another bacterium effectively used in controlling sheath blight and blast of paddy, wilt diseases of ridgeworm, and banana. Suppressive soils. Many soil-borne pathogens, such as Fusarium oxysporum, causing vascular wilts. Geomenorrhaeus graminis, causing take-all of wheat, Pythium spp, causing damping of, and Heterodera avanae, oat cyst nematode, develop well and cause severe diseases in some soils, known as conducive soils, whereas they develop much less and cause much milder diseases in other soils, known as suppressive soils. The mechanisms by which soils are suppressive to different pathogens may involve biotic and or abiotic factors and may vary with the pathogen. They operate primarily by the presence in such soils of one or several microorganisms antagonistic to the pathogen. Many kinds of antagonistic microorganisms have been found to increase in suppressive soils, such as Trichoderma, Penicillium, and Sporidesmium, or bacteria Pseudomonas, Bacillus and Streptomyces. Use of resistant varieties. Grow varieties that have both vertical, initial inoculum limiting, and horizontal, rate limiting, resistance, and most resistant varieties have both type of resistance. Many of them carry only one or a few genes of vertical resistance and an unspecified number of genes of horizontal resistance. 2226. Such varieties are resistant only to some of the races of pathogen, and if the pathogen is airborne, a new race can be brought in easily, as happens with cereal rusts, powdery mildews and Phytophthora infestans. The new race virulent to the resistant variety may appear and become widespread in this way. 5 chemical methods. Fungicides. Definition. The word fungicide originated from two Latin words, vis, fungus, and sidu. The word sidu means to kill. Thus the fungicide is any agency chemical which has the ability to kill the fungus. According to this meaning, physical agents like ultraviolet light and heat should also be considered as fungicides. However, in common usage, the meaning is restricted to chemicals only. Hence, fungicide is a chemical which is capable of killing fungi. Fungistat. Some chemicals do not kill the fungal pathogens, but they simply arrest the growth of the fungus temporarily. These chemicals are called fungistat, and the phenomenon of temporarily inhibiting the fungal growth is termed as fungistasis. Antisporulant. Some other chemicals may inhibit the spore production without affecting the growth of vegetative hyphas and are called as antisporulant. Evenhoff, the antisporulant and fungistatic compounds do not kill the fungi. They are included under the broad term fungicide because by common usage, the fungicide has been defined as a chemical agent which has the ability to reduce or prevent the damage caused to plants and their products. Characters of an ideal fungicide: it should have low phytotoxicity, it should have long shelf life stability during dilution, it should be less toxic to human being, cattle, earthworms, microorganisms, etc. It should be a broad spectrum in its action. Fungicide preparation should be ready for use. It should have compatibility with other agrochemicals. It must be cheaper. It should be available in different formulations. It should be easily transportable. Methods of applications of fungicides: spraying and dusting. Most important method of applying chemicals on the aerial plant parts: foliar application, which are exposed to different pathogens. 2226. Different chemicals, particularly fungicides, are sprayed either as protective, curative or post-symptom treatments. These chemicals provide a continuous covering on the vulnerable plant surfaces and do not allow the plant pathogens, particularly fungi, to invade them. They also eradicate the already established infections and reduce the secondary inoculum. Different equipments are available for high, low and ultra-low volume sprays in the field. Fungicides generally used as sprays are mancozeb, carbendazim, dodine, etc. Sulfur and copper fungicides can also be dusted on the crops for controlling some diseases under high humidity conditions. The soil treatment methods: soil drenching. Vegetables, ornamentals and trees are attacked by many pathogens which are present in the soil, like fusarium and verticillium and some bacteria. Different chemicals are used as soil drench, dust or granules inside the soil at the time of planting of the nursery or seedlings to control damping of seedling blight, crown and root rot and many other soil-borne diseases. These chemicals can also be applied with the irrigation water, wherever the irrigation is possible, especially with the drip irrigation system. Fungicides such as Captan, Metlaxial, PCNB and Chloronub, etc. can be used as soil treatment to overcome above diseases. Fumigation. Most important method for controlling the nematodes and other soil-borne diseases, and chemicals thus used are known as fumigants. Fumigants like formalin, chloropicrin, methylbromide, diazomate and methampsodium are now being used as fumigants in plant protection programs. These chemicals are used as volatile or in gaseous form in the soil and are useful against various groups of organisms like nematodes, insects, fungi, certain bacteria and weeds. Fumigations are normally done in nursery areas and in glass houses. The fumigant is applied to the soil and covered by thin polythene sheets for 5 to 7 days and removed. For example, formaldehyde is applied at 400 ml 100 sq.m. The treated soil was irrigated and used 1 or 2 weeks later. Wappam is normally sprinkled on the soil surface and covered. Volatile liquid fumigants are also injected to a depth of 15 to 20 cm, using subsoil injectors. Seed treatment. Using fungicides on seed is one of the most efficient and economical methods of chemical disease control. Now, the systemic fungicides are impregnated into the seeds to eliminate the deep-seated infection in the seeds. The seed dressing chemicals may be applied by I. Dry treatment, 2. Wet treatment and 3. Slurry. I. Dry seed treatment. 20 to 27. In this method, the fungicide adheres in a fine film on the surface of the seeds. A calculated quantity of fungicide is applied and mixed with seed using machinery specially designed for the purpose. The fungicides may be treated with the seeds of small lots using simple rotary seed dresser, seed treating drum, or of large seed lots at seed processing plants using grain treating machines. Normally in field level, dry seed treatment is carried out in dry rotary seed treating drums which ensure proper coating of the chemical on the surface of seeds. In addition, the dry dressing method is also used in pulses, cotton and oil seeds with the antagonistic fungus like Trichoderma viride by mixing the formulation at the rate of 4 g/kg of the seed. E.g. dry seed treatment in paddy. Mix a required amount of fungicide with required quantity of seeds in a seed treating drum or polythene-lined gunny bags, so as to provide uniform coating of the fungicide over the seeds. Treat the seeds at least 24 hours prior to soaking for sprouting. Any one of the following chemical may be used for treatment at the rate of 2 g/kg, Thiram or Captan or Carboxin or tricyclazole. 2. Wet seed treatment. This method involves preparing fungicide suspension in water, often at field rates, and then dipping the seeds or seedlings or propagative materials for a specified time. The seeds cannot be stored and the treatment has to be done before sowing. This treatment is usually applied for treating vegetatively propagative materials like cuttings, tubers, combs, sets, rhizomes, bulbs, etc., which are not amenable to dry or slurry treatment. Seed dip seed soaking for certain crops. Seed soaking is essential. Seeds treated by these methods have to be properly dried after treatment. The fungicide adheres as a thin film over the seed surface which gives protection against invasion by soil-borne pathogens. E.g. seed dip treatment in paddy. Prepare the fungicidal solution by mixing any of the fungicides with carbendazim or pyroquinone or tricyclazole at the rate of 2 g/liter of water and soak the seeds in the solution for 2 hrs. Drain the solution and keep the seeds for sprouting. E.g. seed dip treatment in wheat. Prepare 0.2% of carboxin, 2 g/liter of water, and soak the seeds for 6 hours. Drain the solution and dry the seeds properly before sowing. This effectively eliminates the loose smut pathogen, Ostiligo nudacritici. Seedling dip. Root dip. The seedlings of vegetables and fruits are normally dipped in 0.25% copper oxychloride or 0.1% carbendazim solution for 5 minutes to protect against seedling blight and rots. Rhizome dip. The rhizomes of cardamom, ginger and turmeric are treated with 0.1% emicin solution for 20 minutes to eliminate rot-causing pathogen present in the soil. Set dip. Sucker dip. The sets of sugarcane and tapioca are dipped in 0.1% emicin solution for 30 minutes. The suckers of pineapple may also be treated by this method to protect from soil-borne diseases. 20 to 28. 3. Slurry treatment, seed pelleting. In this method, chemical is applied in the form of a thin paste. Active material is dissolved in small quantity of water. The required quantity of the fungicide slurry is mixed with the specified quantity of the seed so that during the process of treatment slurry gets deposited on the surface of seeds in the form of a thin paste which later dries up. Almost all the seed processing units have slurry treaters. In these slurry treaters, the requisite quantity of fungicides slurry is mixed with specified quantity of seed before the seed lot is bagged. The slurry treatment is more efficient than the rotary seed dressers. E.g. seed pelleting in ragi. Mix 2.5 g of carbendazim in 40 ml of water and add 0.5 g of gum to the fungicidal solution. Add 2 kg of seeds to this solution and mix thoroughly to ensure a uniform coating of the fungicide over the seed. Dry the seeds under the shade. Treat the seeds 24 hrs prior to sowing. Special method of applications. 1. Trunk application. Trunk injection. It is normally adopted in coconut gardens to control Tanjavur wilt caused by Ganoderma lucidum. In the infected plant, a downward hole is made to a depth of 3 to 4, at an angle of 45. At the height of 3 from the ground level with the help of an auger. The solution containing 2 g of ore often in soil and 1 g of copper sulfate in 100 milliliters of water is taken in a saline bottle and the bottle is tied with the tree. The hose is inserted into the hole and the stopper is adjusted to allow the solution in drops. After the treatment, the hole is covered with clay. 2. Root feeding. Root feeding is also adopted for the control of Tanjavur wilt of coconut instead of trunk application. The root region is exposed, actively growing young root is selected and given a slanting cut at the tip. The root is inserted into a polythene bag containing 100 milliliters of the fungicidal solution. The mouth of the bag is tied tightly with the root. 3. Pseudostem injection _ This method is very effective in controlling the aphid vector, Pentalonia nigronervosa, of bunchy top of banana. The banana injector is used for injecting the insecticide. 4. Comb injection. It is an effective method used to control Panama wilt of banana caused by Fusarium oxysporum FSP. Cubans. Capsule applicator is used for this purpose. It is nothing but an iron rod of 7 mm thickness to which a handle is attached at one end. The length of the rod is 45 cm and an iron plate is fixed at a distance of 7 cm from the tip. The comb is exposed by removing the soil and a hole is made at 45 angle to a depth of 5 cm. 1 or 2 gelatin capsules containing 50 to 60 mg of carbendazim is pushed in slowly and covered with soil. Instead of capsule, 3 milliliters of 2% carbendazim solution can also be injected into the hole. 20 to 28. Tree wound treatment. Fruit plants are often prone to cuts and wounds during the dormant period when they are pruned. The exposed portion of the plant is first sterilized by swabbing it with antiseptic solution of either sodium hypochlorite or ethyl alcohol. Finally, the entire wounded portion is painted with permanent tree wound dressing such as lanolin paste, chalk paste or Bordeaux paste paint. Post-harvest treatment. Fruits and vegetables are largely damaged after harvest by fungi and bacteria. Many chemicals have been used as spray or dip or fumigation. Post-harvest fungicides are most frequently applied as aqueous suspensions or solutions. Dip application has the advantage of totally submerging the commodity so that maximum opportunity for penetration to the infection sites. Systemic fungicides, particularly thibendazole, benomil, carbendazim, metlaxial, Fostii have been found to be very effective against storage diseases. In addition, dithiacarbamates and antibiotics are also applied to control the post-harvest diseases. Wrapping the harvested products with fungicide-impregnated wax paper is the latest method available. Threat of UB99. Wheat rusts have been very imperative diseases of wheat worldwide. The large-scale cultivation of wheat carrying a single gene Senior 31 for protection against stem rust, Puccinia graminis FSP triticea, over a large area proved to be a primer for the development of a new race virulent on this gene. Initially, this gene, used in breeding at Simith, was selected as it was found allied with resistance to other two rusts like yellow, year 9, and brown rust, LR26. Achieving triple resistance with one gene was very striking for wheat breeders and this gene was used in breeding programs all over the world. This gene was further found to be also linked with other positive traits like enhanced yield and adaptability over an array of environments. With the adoption of Senior 31 bearing cultivars, grown all over the world, the incidence and occurrence of stem rust became meager and sporadic. Thus, breeders stared to view stem rust of wheat as less of a threat. In such circumstances, it was anticipated that sooner or later the pathogen may head back and this gene may lose its effectiveness. This was what precisely happened when a new race, popularly termed as UB99, emerged in highlands of Kenya and threatened wheat cultivation in the world. Integrated disease management. Integrated disease management, IDM, came under focus in 1960s when chemicals, especially fungicides and insecticides, came under the attack from environmentalists due to the overuse of chemicals that created the problems of environmental pollution, chemical residues in foodstuff, land, water and air, and the associated health hazards. 20-29. It focused on the other methods of disease control. It involved cultural, biological, epidemiological and alternative means to achieve disease control. Nowadays, there is an emphasis on disease management rather than on control. Definition of IDM. Disease management system that in the context of associated environment and population dynamics of microorganisms, utilizes all suitable techniques and methods in a manner as compatible as possible and maintains the disease below economic level. In general, it is the integration of all possible and suitable management techniques for the control of diseases. The practices which need to be avoided in IDM are indiscriminate use of fungicides, monoculture and growing of susceptible cultivars. Integrated disease management ensures the proper management of soil health, use of healthy seeds and planting material, application of fungicides when required, field sanitation, cultural practices which suppress the disease, use of biocontrol agents and growing resistant plant genotypes. Different approaches of integrated disease management system. The combined control approach. It is a combination of control methods like adjustment in sowing time, seed treatment, use of resistant variety, chemical spray schedule, etc. This type of IDM is widely practiced as a package of practice where the occurrence of disease is certain and sure. The surveillance-based approach. It is an advanced IDM approach based on crop health monitoring and surveillance, and takes into account the economic threshold levels or economic damage levels. Advanced integrated disease management system. It involves the high input technology like computer-supported forecasting, remote sensing, scouting, multiple pathogen thresholds, information on life cycle of pathogens, epidemiology of diseases, environmental factor and knowledge-based decision making. Need for IDMO. The crop yield losses, on field and during post-harvest period, caused by pests, diseases and weeds are of paramount importance. The crop losses due to pests, diseases and weeds are approximately assessed to be ranging between 10 to 30% of crop productions. Chemical pesticides have reduced crop losses in many situations, but even with a substantial increase in pesticide use, the overall proportion of crop losses and the absolute value of these losses from diseases appear to have increased over time. Nonetheless, an increase in pesticide use still appears to be profitable. Increased monoculture, reduced crop diversity and rotation, and use of herbicide have all increased vulnerability to diseases as well. 20-30. Pathogens tend to develop resistance to pesticides, requiring higher use to sustain production. Inappropriate and excessive pesticide use led to increased and unnecessary disease outbreaks and additional disease loss because of the inadvertent destruction of natural enemies of diseases, disease resistance and resurgence of secondary diseases. Ultimately, overuse of pesticides can reduce food production. Proponents with varying perspective on chemicals agree threat integrated disease management, IDM, must be science-based and economically viable for farmers. The emphasis is on disease problems and preventing them from reaching economically damaging levels. Host plant resistance, natural plant products, biopesticides, natural enemies and agronomic practices offer a potentially viable option for IDM. They are relatively safe for the non-target organisms and humans. Biotechnological tools such as marker-assisted selection, genetic engineering, and wide hybridization to develop resistant crop cultivars will have a great bearing on future disease management programs. Disease modeling, decision support systems, and remote sensing would contribute to scaling up and dissemination of IDM strategies. Main components of integrated disease management, IDM. Host resistance. Induced systemic resistance. 3. Genetically improved plants. Cultural practices. Physical methods. Plant nutrition. Biological control. Use of pesticides of plant origin. Judicious use of chemicals. 1. Host resistance. Resistant varieties can be the simple, practical, effective and economical method of plant disease control. Apart from ensuring protection from diseases, they can also save time, money and energy spent on other methods of control and avoid environmental pollution with chemicals. They are the only practical method of controlling such diseases as wilts, rusts and others caused by viruses in which chemical control is very expensive and impractical. In low-value crops, where other methods are often too expensive, development of varieties resistant to common and important diseases can be an acceptable recommendation for the farmers. 20-30. Disease resistance in plants is also governed by their genetic constitution and can be monogenic, oligogenic or polygenic. Advantages of host plant resistance. No adverse effect on environment and man, rather the resistant cultivars put a constant and cumulative effect on pathogen. Host plant involves no extra cost to the farmers and does not require inputs and application skills. Disadvantages of host plant resistance. The development of pathogen-resistant variety takes 5 to 10 years. Host plant resistance can put a selection pressure on pathogen to the extent that it may lead to the evolution of new biotypes of pathogen. Introduction of varieties with resistance to one pathogen leads to the emergence of new pathogen problem because of the absence of competition from the key pathogen. Induction of host resistance. Plants actively respond to a variety of environmental stimuli, including gravity, light, temperature, physical stress, water and nutrient availability. Plants also respond to a variety of chemical stimuli produced by soil and plant-associated microbes. Such stimuli can either induce or condition plant host defense through biochemical changes that enhance resistance against subsequent infection by a variety of pathogens. Induction of host defense can be local and or systemic in nature depending on the type, source, and amount of stimuli. The systemic acquired resistance, SAR, is mediated by salicylic acid, SA, a compound which is frequently produced following pathogen infection and typically leads to the expression of pathogenesis-related, PR, proteins. These PR proteins include a variety of enzymes, some of which may act directly to lyse the invading cells, reinforce cell wall boundaries to resist infections, or induce localized cell death. Whereas, the induced systemic resistance, ISR, is mediated by jasmonic acid, JA, and or ethylene, which are produced following applications of some non-pathogenic rhizobacteria. Interestingly, the SA and JA-dependent defense pathways can be mutually antagonistic, and some bacterial pathogens take advantage of this to overcome the SAR. Pathogenic strains of Pseudomonas syringae produce coronatine, which is similar to JA, to overcome the SA-mediated pathway. Because various host resistance pathways can be activated to varying degrees by different microbes and insect feeding, it is plausible that multiple stimuli are constantly being received and processed by the plant. Thus the magnitude and duration of host defense induction will likely vary over time. 3. Genetically improved plants. 20-31, genes from plants, microbes and animals can be combined and introduced into the living cells of other organisms, and the organisms that have genes from other species inserted into their genome are called transgenics. Production of disease-resistant transgenic plants has been achieved by this method. Certain genes are inserted into plant genome that confer resistance to pathogens such as viruses, fungi and insects. These transgenic plants reduce the pesticide use and thereby provide environmental benefits while reducing farmers' cost. Genetically modified plants are generally used to control the viral diseases. E.G. A transgenic papaya cultivar, Rainbow, has been developed which is resistant to papaya ring spot virus in the US. Integration of different cultural practices. Different cultural practices like crop rotation, mulching, tillage, different soil amendments, soil solarization, soil sterilization, change in date of sowing, plant spacing etc. when applied alone are able to control diseases up to some extent, but when these cultural practices are combined with each other, they not only control the diseases but also increase the yield of crops. The intercropping of maize and sorghum with peppers serves as barriers against the aphid vectors of pepper veinal mottle virus and reduces the virus spread. Soil solarization for 40 days along with the addition of cabbage, cauliflower, broccoli and sarsen leaf residues controlled the gladiolis wilt, Fusarium oxysporum F.sp. gladioli, by 74.6%, whereas soil solarization, for 40 days, alone reduced the gladiolis wilt by 67.3% compared to the unsolarized control. Physical methods of disease control. Solar heat treatment of the water-soaked wheat seed in May to June for 5 to 6 hours provides good control of loose smut of wheat. Most of the post-harvest diseases can be avoided by irradiation, refrigeration, controlled atmosphere storage etc. Soil solarization has been used to control soil-borne diseases caused by otherwise difficult to control fungi, E.g., Rhizoctonia solni, Fusarium spp, Sclerotium etc. In this the soil beds are first irrigated and then covered with thin, 20 transparent mulch in the months of April, May and June. It raised the soil temperatures in some cases up to 500 C, which is deleterious to many plant pathogens in the soil. It has been used in raising disease-free nursery in tropical and subtropical climatic areas. It also provides excellent weed control. Hot water treatment of cabbage seed at 520 C for 15 to 20 minutes controls black rot disease, caused by Xanthomonas campestris PV campestris. Plant nutrition 2231. The nutrition of crop plants has direct effect on the diseases, and is an important component of integrated disease management, IDM. Both deficient and overnourished plants invite high incidence of diseases as well as loss in yield and quality of produce and products. The amount, proportion, time and method of application of fertilizers affect the metabolism of plants and thus occurrence and severity of diseases. Fertilization with both P and K significantly reduces the leaf rust damage and powdery mildew infection in wheat. The deficiency of macronutrients may also affect the incidence of many diseases. Potassium, K, plays an important role in survival of crop plants under environmental stress conditions. Potassium also affects the reaction of plants to pests or diseases by having direct effect on the pathogen number, development, multiplication, survival, vigor and length of life cycle. 7. Biological control. Biocontrol agents are used as a core component of integrated disease management system. The science and art of using living organisms as biocontrol agents is an important component of environment-friendly disease management procedures. These biocontrol agents are of enormous value in integrated diseases management for sustainable agriculture, where they often replace the need of fungicides. The biocontrol agents either suppress the pathogen growth either by the antibiotic production, hyperparasitism or by competition. Various biocontrol agents used in control of various diseases are Bacillus subtilis, Pseudomonas fluorescens, Gliocladium spp, Trichoderma spp, Chaetermium globosum, Pseudomonas ceapacia, Bacillus cereus, Agrobacterium radiobacter etc. Trichoderma viride is the most important and versatile biocontrol agent used for the control of a number of plant pathogens like Rhizoctonia solni and Sclerotium rolfsi, which are otherwise difficult to control by other methods. Similarly, Fusarium litriticum has been used to cover primary wounds of apricot for avoiding the canker disease caused by Eutypa armeniaceae. Ampelomyces quisqualis and Daluca spp hyperparasitize powdery mildew and rust fungi, respectively, and therefore exploited for their biological control. Agrobacterium radiobacter K-84 strain has been used against crown gall disease world over. Use of pesticides of plant origin. Pesticides of plant origin are derived from plant parts, and their genes are also used to transform crops to express resistance to insect, fungal and viral attack. The plant parts and their extracts with antifungal properties play an important role in plant disease management. Plants with pest-killing properties have been recorded as early as Rig Veda in India. 2232, garlic, Allium sativum, has a long history of reputed value and actual use for its medicinal, antimicrobial and pesticidal properties. The growth of Rhizoctonia solni can be reduced with ethanolic extracts of Eucalyptus sp, Canopodium ambrosiades, Lipia alba, Igel marmelos and Cestrum diurnum leaves. The seed extract of Piper nigrum was found to be effective against Arbata ticola. 9. Judicious use of fungicides. Chemicals have been used successfully to combat the ravages of these diseases for many years. Fungicides with different modes of action like protective, broad-spectrum fungicides, post-infection activity, EBI, pre-symptom and post-symptom, benzimidazoles and triazoles, may be used for controlling a wide array of plant diseases ravaging various crops. The overuse of these chemicals resulted in water pollution, residues on food and fruit crops, effect on non-target organisms and development of resistance in pathogens against the chemicals have drawn the attention toward the rational use of fungicides by including monitored control strategies and cultural practices. Types of integrated disease management. I. Integration of cultural and chemical control. The integration of chemicals and cultural practices, including improved cultivars, has resulted in a continuous supply of fresh watermelons, reduced diseases caused by Caletotrichum laganarium, Pseudomonas syringae PV, lacrimans and Pseudoperonospora cubensis. The covering the tomato nursery seedlings with nylon net for 25 to 30 days plus 4 sprays of monocrotophos at 10 days intervals after transplanting delayed the spread of tomato leaf curl virus for 3 to 5 weeks and increased tomato yields. 2. Integration of chemical and biological control. Biocontrol agents such as Pseudomonas fluorescens, Trichoderma viride, T. herzianum, Bacillus subtilis, Pseudomonas putida, PCA asia, Thalaromyces flavus, and Agrobacterium radiobacter strain K84 etc. can be used with integration of chemicals for the effective control of certain diseases. 3. Integration of resistance, cultural, biological and chemical control. The integration of cultural practices, crop rotation, good farm hygiene procedures, quarantine, fertilizers, soil fumigation and solarization, pesticides, fungicide transplant dips, soil drench, soil incorporations, seed treatments, trace elements and surfactants, resistant cultivars and biocontrol agents are used for the control of clubroot, Plasmodiophora brassicae, of vegetables. Advantages. Integrated approach integrates preventive and corrective measures to keep pathogen from causing significant problems, with minimum risk or hazard to human and desirable components of their environment. Some of the benefits of an integrated approach are as follows. 2233 promotes sound structures and healthy plants, promotes the sustainable bio-based disease management alternatives, reduces the risk associated with management by encouraging the adoption of more ecologically benign control tactics, reduces the potential for air and groundwater contamination, protects the non-target species through reduced impact of plant disease management activities, reduces the need for pesticides and fungicides by using several management methods, reduces or eliminates issues related to pesticide residue, reduces or eliminates re-entry interval restrictions, decreases workers, tenants and public exposure to chemicals, alleviates concern of the public about pest and pesticide related practices, maintains or increases the cost effectiveness of disease management programs, adoption implementation of IDM. IDM is multidisciplinary approach seems promising to manage diseases effectively by inclusive amalgamation of cultural, physical, biological and chemical strategies. However, the boon in the knowledge-centric array of IDM ironically proves a bane on reaching the knowledge-poor and resource-deficient farmers at the implementation stage. Socio-economic and educational status of farmers have had a lot of bearing on the adoption success of the IDM program. More worrisome are environmental insensitivities of a majority of farmers, either due to lack of knowledge or desperate economic situations, mainly the rural indebtedness. Unprofessional and unstructured farm mechanism of small land holding makes the socio-economic situation of most of the Indian farmers unenviable. Under such situations, the expectation that knowledge-intensive IDM should succeed appears far-fetched. Under such compelling situations, most of the IDM implementation efforts in the country have been rather sporadic in terms of temporal and spatial. As per current estimates, only 5% of the cropped area in the country is under IDM. 2235. 5. Biotechnology in plant pathology. Diverse approaches in plant disease management have very much been influenced by the recent advances in molecular biology. Many biotechnological tools and techniques have been developed by using different plant pathogens as experimental materials. It has provided way to understand host pathogen correlation under diverse environment. To give a novel look to this branch of science paradoxically viewed as cut and burn technology, different aspects are as follows. I. Molecular diagnosis of plant pathogens. Conventionally, cultural methods have been employed to isolate and identify potential pathogens. This is a relatively slow process, often requiring skilled taxonomists to reliably identify the pathogen. However, over the last 30 years, several techniques have been developed which have found application in plant pathogen diagnosis. These include the use of monoclonal antibodies and enzyme-linked immunosorbent assay, ELISA, and DNA-based technologies, such as the polymerase chain reaction, PCR, which enable regions of the pathogen's genome to be amplified by several million-fold, thus increasing the sensitivity of pathogen detection. Furthermore, diagnostic PCR has been significantly improved by the introduction of second-generation PCR, known as the real-time PCR. It is now possible not only to detect the presence or absence of the target pathogen, but also to quantify the amount present in the sample. Enumerating the pathogen upon detection is crucial to estimate the potential risks with respect to disease development and provides a useful basis for disease management decisions. The DNA microarray technology, originally designed to study gene expression and generate single nucleotide polymorphism, SNP, profiles, is currently a new and emerging pathogen diagnostic technology and offers a platform for unlimited multiplexing capability. The fast-growing databases generated by genomics and biosystematics research provide unique opportunities for the design of more versatile, high-throughput, sensitive and specific molecular assays that will address the major limitation of the current technologies and benefit plant pathology. Finally, the so far restricted use of robotics to DNA technology will become economically feasible and thus accessible to farmers and will offer the possibility of using single DNA chip as practical tool for the diagnosis of hundreds of plant pathogens. 2. Analysis of molecular variability in plant pathogens. Characterization of genetic diversity in plant pathogens have been made feasible, beyond use of differential hosts, through diverse molecular techniques. Different molecular markers have been used in characterization of genetic diversity of plant pathogens. In most of the cases, these are RAP, RFLP, AFLP, SSR, ISSR, ITS. 2236. The RAP markers have been mostly used for characterization of fungal pathogens, followed by AFLP and its markers. This might be because of their ease and simplicity in use. Mapping of disease resistance genes using DNA markers. Molecular mapping can be used for direct selection of disease resistance genes for the use in plant breeding programs. Commonly used markers are restricted fragment length polymorphism, RFLPs, amplified fragment length polymorphism, AFLPs, simple sequence repeats, SSRs, single nucleotide polymorphism, SNP, with predilection of PCR-based markers. There is very few reports available from India which is required to be strengthened to support marker-assisted selection, MAS, in plant breeding for disease resistance. Marker-assisted pyramiding of disease resistance genes. Marker-assisted pyramiding of disease resistance genes termed as breeding by design can help to control the pathogen which recurrently and rapidly develop their new virulence. Efforts are made in India under Asian Rice Biotechnology Network, ARBN, to pyramid resistance gene against bacterial blight of rice. Rice is among the first crops where marker-assisted pyramiding of disease resistance genes was initiated. Rice varieties developed by using MAS have now been released for commercial cultivation for the first time in India. The variety Amend as improved Pusa Basmati 1 was developed by using conventional plant breeding approach integrated with MAS and two bacterial blight resistance genes XA13 and XA21 incorporated in Pusa Basmati 1. Another variety of rice resistant to bacterial blight was developed in non-basmati type rice in India by using MAS. PCR-based molecular markers were used in a backcross breeding program to introgress three major bacterial blight resistance genes, XA21, XA13 and XA5, into Samba Mashari from a donor line, SS1113, in which all the three genes are present in a homozygous condition. These two reports successfully demonstrate the application of marker-assisted selection for targeted introgression of BLB resistance genes into basmati type and non-basmati types varieties of rice in India. So, there is call for to take up such initiatives for other host pathogen system for control of serious loss-causing diseases. Molecular markers can also help in assaying the germplasm for presence or absence of a particular disease resistance gene. Cloning of disease resistance genes by tagging approaches can identify the function of a specific genes by uncovering a specific pathotype. Transgenics. Development of disease resistance through transgenic research is yet at primitive stage in India. Non-availability of resistance to plant pathogens can be overcome by search and transfer of resistance genes from other sexually incompatible species, which is possible using genetic engineering approach. 2236. It requires to first search for new genes which have broad spectrum resistance to pathogen population present in the region and then following transgenic approach, transferring of resistance to commercial varieties to achieve resistance, working at field level. Disease resistance transgenic have been developed in banana and tobacco by transferring a synthetic substitution analog of a short peptide, mugnin. Meganine is one of the earliest reported antimicrobial peptides from skin secretions of the African clawed frog. The peptide is not stable in its native form and, therefore, researchers modified it to express in foreign plant systems. Tobacco plants transformed with the peptide showed enhanced resistance against Sclerotinia sclerotium, Alternaria alternata and Botrytis cinerea. Transgenic banana plants showed resistance to Fusarium oxysporum fsp, Cubensi and Mycosphaerella musicola. However, it remains to be seen how these plants perform under natural disease conditions. Application of genomics. Genomic has emerged as one of the frontier technologies during this century. Its application in different areas of plant pathology can be enormous in structural, functional or comparative genomics. Using high-throughput genome sequencing technologies, many plant pathogens are being sequenced world over. The massive genome sequence data being generated on different microorganisms can be used for simultaneous detection of multiple plant pathogens. The unique sequence from a wider range of pathogens could be used to develop microarrays for the simultaneous detection of large number of different strains. The probes and primers could be designed for differential detection of pathogens and their characterization at molecular level by using the unique sequence data of the pathogen's DNA. Application of RNA interference. RNA interference, RNI, has emerged as a powerful tool for battling some of the most notoriously challenging diseases caused by viruses, bacteria and fungi. RNI is a mechanism for RNA-guided regulation of gene expression in which double-stranded ribonucleic acid, dsRNA, inhibits the expression of genes with complementary nucleotide sequences. The application of tissue-specific inducible gene silencing in combination with the use of appropriate promoters to silence several genes simultaneously will result in protection of crops against destructive pathogens. RNI application has resulted in successful control of many economically important diseases and pests in plants. The RNI-based technologies have tremendous potential in significantly reducing our reliance on chemical pesticides and thus pave way for the efficient, cost-effective, eco-friendly alternative which will ensure a paradigm shift in pest management in India. 2237. 8. Post-transcriptional gene silencing. The RNA silencing mechanism is also a powerful tool to develop crop species resistant to viruses. The expression of virus-derived sense or antisense RNA in transgenic plants conferring RNA-mediated virus resistance appears to induce a form of post-transcriptional gene silencing, PGTS. It's a nucleotide sequence-specific process that includes mRNA degradation, RNA silencing, an evolutionary mechanism protecting cells from pathogenic RNA and DNA, is viewed as an adaptive immune system of plants against viruses. Several lines of research indicate that RNA silencing can be induced locally and then spread throughout the organisms, and this aspect of the process likely reflects its role in viral defense. Role of nanotechnology. Nanotechnology offers an imperative role in improving the existing crop management techniques. Generally, only a very low concentration of agrochemicals have reached the target site of crops due to leaching of chemicals, degradation by photolysis, hydrolysis and microbial degradation. Hence, repetitive application is needed for effective control causing unfavorable effects such as rapid occurrence of resistance and soil and water pollution. Nano-formulated agrochemicals should be designed in such a way that they hold all necessary properties like effective concentration, with high solubility, stability and effectiveness, time-controlled release in response to certain stimuli, improved target activity and less ecotoxicity with safe and easy mode of delivery. Therefore, an urgent need is to evaluate and develop natural, biodegradable, and environment-safe nano-formulated compounds. 2237. 6. Host plant resistance. A physiological deviation from the normal functioning of the organism, i.e., the crop plant, caused by pathogenic organisms is a disease and may be caused by fungi, bacteria or viruses. The inherent ability of an organism, i.e., the crop plant, to resist or withstand the pathogen is called resistance. Disease resistance commonly met with in the plant kingdom relative in nature, total immunity being too rare. Its hereditary transmission from parent to offspring is essentially Mendelian, but often polygenic. Resistant varieties can be the simplest, practical, effective and economical method of plant disease control. The use of resistant varieties can not only ensure protection against diseases but also save the time, energy and money spent on other measures of control. In addition to these advantages, resistant varieties, if evolved, can be the only practical method of control of such diseases as viruses, phytoplasmas wilts, and rusts etc. in which chemical control is very expensive and impractical. In crops of low cash value, chemical and other methods of control are often too expensive to be applied. In such crops, development of varieties resistant to important diseases can be an acceptable recommendation for the farmer. Pathogenicity is the ability of a pathogen to attack a host. Pathogenicity includes virulence and aggressiveness. Virulent strains of pathogen cause much severe symptoms of the disease and they carry the virulence gene that enables it to attack a particular host genotype. Virulence is due to the action of one or a few genes. An aggressive strain of a pathogen causes severe disease on all the host genotypes which they are able to attack and aggressiveness is polygenically inherited. Host-pathogen relationship. A disease is the result of an interaction of genes governing resistance in the host with those governing pathogenicity only in the on the pathogen. Genotype D of resistance, the host for of resistance, a crop to but a physiological also upon the race genotype of the pathogen depends not of the pathogen for virulence or aggressiveness. Resistance that floor there, 1942, are in the at least proposed corresponding host. To there the alleles is gene for gene locus at corresponding in locus the pathogen controlling hypothesis, gene, VV, resistance susceptibility for pathogenicity controlling according only to virulence in which, the in pathogen. For aggressiveness. The every host leads, RR, gene it means 2 and 4. 2 out alleles of at between 1 combination genetic. The 4 possible interactions of gene for gene these alleles, relationship requires studies expression of resistance, the demonstration of both the host and the pathogen. 2238. Vertical resistance, VR, and horizontal resistance, HR. Van der Planck, 1960, has discussed the whole issue of disease resistance in a different perspective. He calls the unstable and often complete type of resistance as vertical resistance and the more stable but somewhat incomplete resistance as horizontal resistance. If resistance to some races of a pathogen is more than 2 races, it is called vertical resistance. It is also called perpendicular resistance, physiological resistance, seedling resistance. Hypersensitivity, race-specific resistance or qualitative resistance. As it is conditioned by one or a few genes, it is called major gene or monogenic of oligogenic resistance. Resistance to more than one race of the pathogen or to many or all races of the pathogen is called horizontal resistance. It is non-specific resistance governed by polygenes. It is severally termed as non-specific, general, polygenic, minor gene, mature plant, adult, quantitative resistance, partial or field resistance or tolerance. HR causes reduction in the number and rate of sporulation of the pathogen on the host and slows down the infection rate. HR includes tolerance, slow development of disease, escape and exclusion mechanisms besides hypersensitive reaction. Differences between vertical and horizontal disease resistance * detectable by analysis of variance of a suitable experiment. 2238. Pathotype specificity, race specific, race non-specific. Nature of gene action J oligogenic, polygenic, rarely oligogenic. Response to pathogen usually hypersensitive resistant response. Phenotypic expression qualitative quantitative. Stage of expression seedling to maturity expression increase as plant mature adult plant selection and evaluation relatively easy relatively difficult risk of boom and burst present rarely durable absent durable suitable for 1 host 2 pathogen annual but not perennial immobile pathogen e.g. soil pathogen but for mobile airborne pathogen both annuals and perennial all pathogen. 2248. Need for specific deployment of resistant varieties critical for success with mobile pathogen. None need for other control measures likely much less likely host pathogen interaction present absent efficiency highly efficient against specific races variable but operates against all races. 2249. Vertical resistance to specific races is generally governed by a single, monogenic, dominant gene or by a few dominant genes. Some of these genes may be multiple alleles as in leaf rust gene LR2 that accords resistance to Puccinia recondite triticale. In that locus, 4 genes designated as LR2A, LR2B, LR2C and LR2D are present and are tightly linked. Each of these genes accord resistance to a different spectrum of races and hence can be differentiated from one another. Such multiple alleles exist on senior 9 locus of wheat for P. Graminease triticea and gene PK in rice for resistance to Pyracularia grycea. The tight linkage between the multiple alleles permits an efficient transfer of all these genes in one attempt. Horizontal resistance, HR, reduces the rate of disease spread and is evenly spread against all races of the pathogen. The low terminal disease severity in HR is assumed to result from polygenic resistance. Morphological features such as size of stomata, stomatal density per unit area, hairiness, waxiness and several others influence the degree of resistance expressed. Partial resistance, dilatory resistance, lasting resistance are some other terms coined for denoting horizontal resistance. The phenomenon of slow rusting manifested as lesser number of pustules per unit leaf area, smaller size of eurydosory and increased latent period in some wheat cultivars is a typical example of this type of resistance. Although it is preferable to use varieties that have both vertical and horizontal resistance, most of the resistant varieties carry only one or few, 2 or 3, major genes of vertical resistance. If varieties are resistant only to some of the races of pathogen and if the pathogen is airborne, then new races evolve easily, as happens with cereal rusts, the Poderi mildew and Phytophthora infestans. Appearance of new races lead to breakdown of resistance of the popular, ruling genotype. As a result, varieties with vertical resistance need to be replaced at frequent intervals. Boom and burst cycle. In varietal improvement programs, it is easy to incorporate the monogenic vertical resistance genes. But the success of exploiting the monogenic host resistance invariably does not last long. 2250. Acreage frequency increases. Breeder produces increased cultivar with host epidemic pathogen virulence new resistancy detected. Acreage frequency decreases. Whenever a single gene-based resistant variety is widely adopted, the impact would be the arrival of new matching pathotypes. These pathotypes soon build up in population to create epidemics and eventually the variety is withdrawn. This phenomenon is generally called boom and burst. To avoid the implications of boom and burst phenomenon, use of durable host resistance is advocated in several crops. Durable resistance remains effective even though it may be widely grown over a long period of time, in an environment that favors the disease. For example, oat variety, red rust proof is still resistant against crown rust even after 100 years. Wheat varieties, Thatcher and Lee have withstood stem rust for 55 and 30 years, respectively. Capelli dispres expresses at adult stage, a moderate resistance to yellow rust and this has been maintained for the last 20 years. 2 of the genes like LR34 for resistance of leaf rust and senior 2 for resistance to stem rust have been recognized for durability. Mechanism of disease resistance or nature of disease resistance. Disease resistance is governed by several inbuilt mechanisms of the host, plants against infection by the pathogen. They are disease escape, disease endurance or tolerance and true resistance. A disease escape. It is a prevention mechanism that causes the host to escape pathogenic infection. Early or late maturity of the crop may prevent physical contact of the pathogen with the host. Mechanical and anatomical barriers such as thick cuticle, waxy bloom on leaves and stem, stomatal regulation prevent penetration of spores. Ergot, a fungal disease of inflorescence in cereals caused by Claviceps purpurea does not affect varieties of wheat and barley in which the flowers remain closed until pollination occurs. Erect leaves of barley avoid deposition of spores of Erysiphe graminease triticea in contrast to prostrate leaves. Early maturing varieties of groundnut escape early leaf spot infection, Cercospora arachidicola, and early varieties of wheat escape rust and loose smut infection. A change in planting season has also been successfully employed as a measure of securing escape, E.g., the leaf rust of sugarcane, Puccinia saccharine, in the canal areas of Bombay severely. 2251. Affects cane when planted in June, but is of minor importance or absent in crops sown in October. Disease escape confers pseudo-resistance. Disease endurance. The host after being infected by the pathogen tolerates the infection and suffers less damage. It does not result in any substantial decrease in yield. This is brought about by influence of external factors. It is a well-known phenomenon that plants fertilized with phosphatic and potash manures are more tolerant to disease. This is the case in wheat against rust infection. Rice crops fertilized by silicates are resistant to blast, Pyracularia oryzae, in Japan. Wheat crops fertilized by potash and phosphatic manures are highly tolerant to mildew and rust infection. The fertilizers act indirectly to arrest vegetative growth and promote early maturity, better straw and strengthening tissues to protect the plant which form a bulwark against pathogenic invasion. True resistance. It is the ability of the host plant to resist or withstand the attack of a pathogen. True resistance is inheritable and much less subject to environmental influence. It is specific in character. The basis of resistance may be morphological, functional, structural or protoplasmic. Functional nature of resistance is determined by opening of the stomata, time of opening of flowers and time of maturity, rate of cork formation and cambial activity. Structural characters include the proportion of strengthening tissues, fiber content, nature of middle lamella, corky layers, number and structure of stomata and lenticels and their sizes. Protoplasmic factors controlling resistance are related to cell contents and include acids, tannins, anthocyanins. Chemical constituents and their proportion, antibiotic activity and hypersensitivity present in the plant cells and in addition biological antagonism of the protoplasm of the host and the pathogen. True resistance, however, is of a specific character and is determined by the defense equipment and activities of the plant itself against the parasitic invasion and is therefore not subject to any appreciable modifications by external factors. Methods of breeding for disease resistance. The methods of breeding varieties resistant to diseases do not differ greatly from those adopted for other characters. The following methods are used. Introduction. Selection. Hybridization followed by selection. Backcross method. Induced mutagenesis. Development of multilines and tissue culture techniques. 2251. Introduction. It is a very simple and inexpensive method. Varieties resistant to a particular disease elsewhere may be thoroughly tested in the regions in which they are proposed to be introduced. Their yield performance and disease resistance should be confirmed by large-scale cultivation. It is possible that a variety resistant in one region need not be resistant in another region due to variation in the physiological race of the pathogen or due to a much different agroclimatic condition in the new location. Introductions have served as a useful method of disease control. For example, Ridley wheat introduced from Australia has been useful as a rust-resistant variety. Manila, a rice variety introduced in Karnataka from the Philippines, has tolerance to blast, bacterial leaf blight and sheath blight. Intan, a Javnika-type rice variety introduced in Karnataka from Indonesia is highly resistant to blast. Munal, a rice variety introduced in West Bengal from the USA is tolerant to blast, bacterial leaf blight and leaf folder, pest. Some of Eri rice varieties such as IR20, IR.24, IR.28, IR.34, IR.36 and IR.50 possess resistance to one or more diseases. Early varieties of groundnut introduced from USA have been resistant to luff spot, Cercospora arachidicola. Kalyan Sona and Sonalika wheat varieties originated from the segregating materials introduced from Simit, Mexico and were rust resistant. Introductions also serve as sources of resistance in breeding programs. For example, African pearl millet, P. americanum, introductions have been used for developing downy mildew-resistant male sterile lines, TIFT23 acetoplams, for use in hybrid pearl millet production. This is an important development in the hybrid pearl millet programs since the original male sterile lines TIFT23A and 23D2 were extremely susceptible to downy mildew. The introduction of Co475 variety of sugarcane in Mumbai has conquered red rot but brought in leaf rust and whip smut to the fore. Selection. This is better method than introduction and has more chances of success in obtaining disease-resistant plants. The work of selection is carried out either in the naturally infected fields under under artificially inoculated conditions. The resistance in such individuals will occur in nature by mutation. To ensure the resistant character of a plant, large population of crop plant may be exposed to the attack of pathogen under artificial conditions and the non-infected plants may be chosen. Suvarnamodan rice of Kerala is a pure line of ARC 11,775 and is highly tolerant to blast. Sugandh of Bihar is a selection from Basmati rice of Odisha tolerant to bacterial leaf blight. Rice varieties Sudha, Bihar, Sabita, Nalini, West Bengal, Patel 85, Madhya Pradesh, Janki, Bihar, improved white ponni, Tamil Nadu, Ambika, Maharashtra, are some of rice selections resistant to one or more diseases. MCU1 cotton, a selection from Co4, is resistant to Kufri red, a potato selection from Darjeeling red round is a disease-resistant variety. 2252. 3. Hybridization. When selection of resistant varieties is not feasible, resistant varieties may be evolved by crossing the susceptible popular variety with resistant wild variety wherein the resistant gene or genes transferred into the genetic makeup of susceptible variety. Very often the F1 from crosses may be resistant but carry the other undesirable qualities of the resistant parent. The bad qualities are removed by several back crossing of F1 with the susceptible parent may ultimately yield a resistant progeny with good agronomic characteristics. Under certain circumstances pedigree or bulk method of selection is followed to obtain a resistant variety. In this method, the crosses are made till F2 population is got. Selections are made in F2 generation for superior genetic traits including disease resistance. By continued self-wing, selections are made through F3 to F5 or F6 generations and the best variety is selected. This method is suited for small grains and beans but unsuited to fruits and vegetables. 4. Backcross method. Backcross method is widely used to transfer disease resistance from wild species. Wild species are rice sources of disease resistance. Interspecific hybridization is made to transfer the gene or genes for resistance to the cultivated species. Resistance to grassy stunt virus from Oryza nivra to O. sativa, late blight resistance from Solanum demissum to cultivated potato, rust resistance from durum to estivum wheat are some of the examples involving interspecific hybridization. Depending upon the number of genes governing resistance and the nature of the gene, whether dominant or recessive, the procedure varies. The number of back crosses to the cultivated species may be 5 to 6. Once the back cross progeny resemble the cultivated parent, then they are self and segregating progeny screened for disease resistance. 5. Induced mutagenesis. While following mutation breeding for disease resistance, a large number of mutation progeny should be produced and screened under artificial epiphytotic condition to select resistant plants. MCU10 cotton, a resistant variety to bacterial blight was evolved in Tamil Nadu by subjecting seeds of a susceptible variety Co4 to gamma rays followed by rigorous screening and selection. 6. Development of multilines. The concept of multilines was first suggested by Jensen, 1952, and developed by Bolog, 1959, for evolving multiline varieties to resist stem rust in wheat. A multiline variety is a composite of genetically similar lines, except that each line possesses a different gene for resistance to the pathogen. Lines that are genetically similar, except for one gene, are called isoline. It is assumed that gene for resistance in each isoline contributes resistance to a separate physiological race or group of races. Genes for disease resistance are transferred by backcrossing from donor varieties to a common disease-susceptible, but agronomically superior, recurrent parent. Isolines are generated differing only in the gene for disease resistance. The isolines are composited to synthesize a multiline variety. 2252. The isolines are maintained for resynthesizing the multiline whenever needed. A multiline variety is composed of a mixture of resistant and susceptible genotypes and provides a buffering effect against rapid development of disease. It will provide resistance or tolerance to a broad spectrum of races of a pathogen. If new races of the pathogen are identified at a later stage, additional isolines resistant to the newly arisen races may be constituted and incorporated. Care should be taken to see that there is uniformity for height, maturity and other features in the multiline. Though multilines provide stability of yield due to reduction of damage by pathogens, the limitations of multiline varieties are that the yield level of multiline varieties is limited to that of the recurrent parent. 4 to 5 years are required to stabilize isogenic lines, and the pathogen may produce new races at a faster rate than the development of a multiline. Multiline varieties have been developed for resistance to stem rust and stripe rust of wheat and crown rust of oats. The first multiline variety in wheat, Memer 60, was developed and released in Colombia to combat the attack of yellow rust. Memer 63 and Memer 65 were resistant to stem rust and stripe rust. Kalyan Sona and Sonalika-based multilines of wheat resistant to different races of rust have been developed in India. 7. Tissue culture technique. Tissue culture techniques to produce somaclonal variation for disease are developed in different crops. Somaclonal variations for disease resistance are reported in Z. maize for Draxlera maydis race T toxin resistance, in Brassica napus for resistance tolerance to Formalingum, early and late blight resistance in potato, Pseudomonas and Alternaria resistance in tobacco, besides smut and rust disease resistance in sugarcane. 2253. 7. Classification of fungicides. Fungicides can be broadly grouped based on their I. Mode of action, 2. General use and 3. Chemical composition. I. Based on mode of action. 1. Protectant. As the name suggests, protectant fungicides are prophylactic in their behavior. Fungicide which is effective only if applied prior to fungal infection is called a protectant. Egg, xenub, sulfur. 2. Therapeutant. Fungicide which is capable of eradicating a fungus after it has caused infection and thereby curing the plant is called chemotherapeutant. Egg carboxin, oxycarboxin antibiotics like orofungin. Usually chemotherapeutant are systemic in their action and affect the deep-seated infection. 3. Eradicant. Eradicant are those which remove pathogenic fungi from an infection coat, area of the host around a propagating unit of a fungus in which infection could possibly occur. 8. Organic mercurials, lime sulfur, dodine etc. These chemicals eradicate the dormant or active pathogen from the host. They can remain effective on or in the host for some time. Based on general uses. The fungicides can also be classified based on the nature of their use in managing the diseases. Seed protectants, e.g. Captan, Thiram, organomercuries carbendazim, carboxin etc. Soil fungicides, preplant, e.g. Bordeaux mixture, copper oxychloride, chloropicrin, formaldehyde, wapum, etc. Soil fungicides, e.g. Bordeaux mixture, copper oxy, for growing plants, chloride, Captan, PCNB, Thiram etc. Foliage and blossom, e.g. Captan, Ferbum, Xenub, protectants mancozeb, chlorothalonil etc. Fruit protectants, e.g. Captan, Menub, carbendazim, mancozeb etc. Eradicants, e.g. organomercurials, lime sulfur, etc. Tree wound dressers, e.g. borax paste, choaptatia paste, etc. Antibiotics, e.g. actidine, griseofulvin, streptomycin, streptocycline, etc. General purpose spray and dust formulations. Based on chemical composition. A. Inorganic chemicals. I. Copper compounds. 2. Inorganic sulfur 2254. 3. Carbonate compounds. 4. Phosphate and phosphonate compounds. B. Mercury fungicides. B. Organic chemicals. I. Organic sulfur compounds or dithiocarbamates. 2. Quinones. 3. Aromatic compounds. 4. Heterocyclic nitrogenous compounds. A. Inorganic chemicals. I. Copper fungicides. The fungicidal action of copper was mentioned as early as 1807 by Prevost against wheat bunt disease, Tilletia caries, but its large-scale use as a fungicide started in 1885 after the discovery of Bordeaux mixture by Millardet in France. The mixture of copper sulfate and lime was effective in controlling downy mildew of grapevine caused by Plasmopra viticola and later, late blight of potato, Phytophthora infestans. Some other copper sulfate preparations later developed were Bordeaux paste, Burgundy mixture and chestnut compound, which are all very effectively used in the control of several plant diseases. In addition, some preparations of copper oxychloride preparations are also used. These are all insoluble copper compounds very successfully used in managing several leaf diseases and seeding diseases in nursery. Some of the important diseases controlled by copper fungicides are listed below. 1. Copper sulfate preparations. Borax mixture. In 1882, Millardet in France, Bordeaux University, accidentally observed the efficacy of the copper sulfate against the downy mildew of grapes caused by Plasmopra viticola. When copper sulfate was mixed with lime suspension, it effectively checked the disease incidence. The mixture of copper sulfate and lime was named as Boilly Bordelaise, Bordeaux mixture. The original formula developed by Millardet contains 5 pounds of QSO4 plus 5 pounds of lime plus 50 gallons of water. The chemistry of Bordeaux mixture is complex and the suggested reaction is QSO4 plus Ca, O, 2Q, O, 2 plus CaSO4. The ultimate mixture contains a gelatinous precipitate of copper hydroxide and calcium sulfate, which is usually sky blue in color. Cupric hydroxide is the active principle and is toxic to 2254. Fungal spores. In metric system, to prepare 1% Bordeaux mixture, the following procedure is adopted. 1 kg of copper sulfate is powdered and dissolved in 50 liters of water. Similarly, 1 kilogram of lime is powdered and dissolved in another 50 liters of water. Then copper sulfate solution is slowly added to lime solution with constant stirring, or alternatively, both the solutions may be poured simultaneously to a third contained and mixed well. The ratio of copper sulfate to lime solution determines the pH of the mixture. The mixture prepared in the above said ratio gives neutral or alkaline mixture. If the quality of the used is inferior, the mixture may become acidic. If the mixture is acidic, it contains free copper, which is highly phytotoxic, resulting in scorching of the plants. Therefore, it is highly essential to test the presence of free copper in the mixture before applied. There are several methods to test the neutrality of the mixture, which are indicated below. Field test: dip a well-polished knife or a sickle in the mixture for few minutes. If reddish deposit appears on the knife sickle, it indicates the acidic nature of the mixture. Litmus paper test: the color of blue litmus paper must not change when dipped in the mixture. 3. pH paper test: if the paper is dipped in the mixture, it should show neutral pH. 4. Chemical test: acid a few drops of the mixture into a test tube containing 5 ml of 10% potassium ferrocyanide. If red precipitate appears, it indicates the acidic nature of the mixture. If the prepared mixture is in the acidic range, it can be brought to neutral or near alkaline condition by adding some more lime solution into the mixture. Bordeaux mixture preparation is cumbersome, and the following precautions are needed during preparation and application. The solution should be prepared in earthen or wooden or plastic vessels. Avoid using metal containers for the preparation, as it is corrosive to metallic vessels. Always copper sulfate solution should be added to the lime solution. Reverse the addition leads to precipitation of copper, and resulted suspension is least toxic. Bordeaux mixture should be prepared fresh every time before spraying. In case the mixture has to be stored for a short time or a day, jaggery can be added at the rate of 100 kg 100 liters of the mixture. Bordeaux mixture is sometimes phytotoxic to apples, peaches, rice varieties like IR8 and match varieties like Ganga Hybrid 3. 2. Copper carbonate preparation: Chaubtatia paste. Chaubtatia paste is another wound dressing fungicide developed by Singh in 1942 at Government Fruit Research Station, Chaubtatia in the Almora district of Uttar Pradesh. It is usually prepared in glass containers or chinaware pot by mixing 800 g of copper carbonate and 800 g of red lead in liter of raw linseed oil or lanolin. This paste is usually applied to pruned parts of apple, pear, 2255, and peaches to control several diseases. The paste has the added advantage that it is not easily washed away by rainwater. Sulfur fungicides. Use of sulfur in plant disease control is probably the oldest one and can be classified as inorganic sulfur and organic sulfur. Inorganic sulfur is used in the form of elemental sulfur or as lime sulfur. Elemental sulfur can be either used as dust or vegetable sulfur, later being more widely used in plant disease control. Sulfur is best known for its effectiveness against powdery mildew of many plants, but also effective against certain rusts, leaf blights and fruit diseases. Sulfur fungicides emit sufficient vapor to prevent the growth of the fungal spores at a distance from the area of deposition. This is an added advantage in sulfur fungicides as compared to other fungi toxicants. E.g., sulfur dust. Sulfur is a contact and protective fungicide, normally applied as sprays or as dust. It is generally used to control powdery mildews of fruits, vegetables, flowers and tobacco. This is also effective against apple scab, Venturi inequalis, and rusts of field crops. Organic compounds of sulfur are now widely used in these days. All these compounds, called as carbamate fungicides, are derivatives of dithiocarbamic acid. Dithiocarbamates, e.g. Zenab, Mainab, e.g. Thiram, Zyram, Mancozeb, Nabam, Wapam, Ferbam. Zenab, against diseases such as early and late blight of potato and tomato, downy mildews and rusts of cereals, blast of rice, fruit rot of chili, etc. Mainab and Mancozeb, these two are protective fungicides used to control many fungal diseases of field crops, fruits, nuts, ornamentals and vegetables, especially blights of potatoes and tomatoes, downy mildews of vines, anthracnose of vegetables and rusts of pulses. Nabam is primarily used for foliar application against leaf spot pathogens of fruits and vegetables. Soil applications were also reported to have a systemic action on Pythium, Fusarium and Phytophthora. It is also used to control algae in paddy fields. Thiram, it is used for seed treatment both as dry powder or as a slurry. It is a protective fungicide also suitable for application to foliage to control Botrytis spp on lettuces, ornamental, soft fruits and vegetables, rust on ornamentals and Venturia perina on pears. It is also effective against soil-borne pathogens like Pythium, Rhizoctonia and Fusarium. Mercury fungicides. Mercury fungicides can be grouped as inorganic and organic mercury compounds. Both the groups are highly fungi toxic and were extensively used as seed treatment chemicals against seed-borne diseases. Ignorance compounds show bactericidal property also. However, due to their residual toxicity in soil and plants and their extreme toxicity nature to animal and human beings, the use of mercury fungicides is being discouraged. In most of the countries, the use of mercury fungicides is banned, and in countries like India, the use of mercury fungicides is restricted only in seed treatment for certain crops. 2255. e.g. mercuric chloride. It is used for treating potato tubers and propagative materials of other root crops. Mercurous chloride is limited to soil application in crop protection use because of its phytotoxicity. B. Organic chemicals. I. Organic sulfur compounds or dithiocarbamates. 2. Quinones. 3. Aromatic compounds. 4. Heterocyclic nitrogenous compounds. I. Heterocyclic nitrogen compounds. Heterocyclic nitrogen compounds are mostly used as foliage and fruits protectants. Some compounds are very effectively used as seed dressers. E.g. Captain, it is a seed dressing fungicide used to control diseases of many fruits, ornamental and vegetable crops against rots and damping of. 2. Quinone fungicides. Quinone are recent naturally in plants and animals and they exhibit antimicrobial activity and some compounds are successfully developed and used in the plant disease control. E.g. diclone. Diclone has been used widely as seed protectant. This is also used as a foliage fungicide, particularly against apple scab and peach leaf curl. Systemic fungicides. Since the late 1960s there has been substantial development in systemic fungicides. Any compound capable of being freely translocated after penetrating the plant is called systemic. A systemic fungicide is defined as fungi-toxic compound that controls a fungal pathogen remote from the point of application, and that can be detected and identified. Thus, a systemic fungicide could eradicate established infection and protect the new parts of the plant. Several systemic fungicides have been used as seed dressing to eliminate seed infection. These chemicals, however, have not been very successful in the cases of trees and shrubs. On the basis of chemical structure, systemic fungicides can be classified as benzimidazoles, thiophthanates, oxatilins and related compounds, pyrimidines, morpholines, organophosphorus compounds and miscellaneous group. I. Oxatilin and related compounds. Carboxin, white avux, it is systemic fungicide used for seed treatment of cereals against bunts and smuts, especially loose smut of wheat. Oxycarboxin, plant wax, it is a systemic fungicide used for the treatment of rust diseases of cereals, pulses, ornamentals, vegetables and coffee. 2256. 2. Benzimidazoles. Benomil, it is a protective and eradicative fungicide with systemic activity, effective against a wide range of fungi affecting field crops, fruits and ornamentals. It is very effective against rice blast, apple scab, powdery mildew of cereals, rose, cucurbits and apple and diseases caused by verticillium and rhizoctonia. It is also used as pre- and post-harvest sprays of dips for the control of storage rots of fruits and vegetables. Carbendazim is a systemic fungicide controlling a wide range of fungal pathogens of field crops, fruits, ornamentals and vegetables. It is used as spray, seedling dip, seed treatment, soil drench and as post-harvest treatment of fruits. It is very effective against wilt diseases especially banana wilt. It controls effectively the sigatoka leaf spot of banana, turmeric leaf spot and rust diseases in many crops. Thibendazole, it is a broad-spectrum systemic fungicide effective against many major fungal diseases. Pathogenic fungal control includes species of Botrytis, Ceratocystis, Cercospora, Caletotrichum, Fusarium, Rhizoctonia, Sclerotinia, Septoria and verticillium. It is also effective for the post-harvest treatment of fruits and vegetables to control storage diseases. Pyrimidines. They include mainly dimethyrimol, atherimol and tririmol which are effective against powdery mildews and some other fungi. They are mainly used for seed and soil treatments. Elakinines. The most important fungicide is metlaxial, which is effective against pythium, phytophthora and several downy mildews. It is sold as Ridomil for use in soil or foliage, and as Apron for use as seed dresser. Metlaxial is the first and one of the best systemic fungicides against oomycetes. Morpholemes. They include the fungicides Dodimorph sold as Meltetox and Tridmorph, sold as Calixin. They are preventive and eradicant foliar fungicides effective against powdery mildews and leaf spots on cereals, ornamentals and tropical plants. Triazoles. They include several excellent systemic fungicides, such as triamphene, balaton, triadimenol, baton, beatanol, Bacor, bautrizol, Inder, and etoconazole, Vanguard. They show a long protective and curative activity against a range of foliar, root and seedling diseases such as leaf spots, blights, powdery mildews, rusts, smuts etc. They are used as foliar sprays and for seed and soil treatments. Most antibiotics are toxic mostly to bacteria, mycoplasma and some fungi. They are generally absorbed and translocated systematically. Antibiotics may act on pathogen or on host directly or after undergoing transformation within the host. Some of the most commonly used antibiotics are the following. 2257. Streptomycin, sold as agrimycin, phytomycin, agristep etc., and used as spray. It is effective against broad range of bacterial pathogens causing spots, blights, wilts, rots etc. It is also used as soil drench to control Xanthomonas sp. and as a dip to seed disinfectant, against bacterial diseases of beans, cotton, crucifers, cereals etc. Tetracyclines are effective against many bacteria and all mycoplasmas. Termycin, oromycin and acromycin are used in disease control. These are injected into trees. Cycloheximide, sold as Actedion is used for many fungal diseases including leaf spots and powdery mildews. New generation fungicides. The process of fungicides discovery has undergone a noteworthy change over the years. After the era of broad-spectrum multi-site and site-specific systemic fungicides, several novel action fungicides of different chemical classes have been developed in the past 2 decades. These are more eco-friendly and used at a much lower dose rates as compared to the earlier compounds. Most noted among these are the strobilurins, cores, derived from Strobilurus tenacellus, a wild mushroom. These are analogues to strobilurins A and have broad range of disease control. Azoxystrobin was the first strobilurin introduced in 1996 and currently 9 strobilurins compounds are available. Other important fungicides introduced for the control of diverse diseases in the last decade are oxazolidinadians, fioxazolone, phenoxyquinolins, quinoxyf1, anilinopyrimidines, cyprodenyl, pyrimethamyl, valinamides, iprovalicarb, bentiocarb. Mundelamides, mundipropamide, phenylpyrrose, fenpiclol, fludiazonil, MBIs, carprapamide, spirocatlamines, spiroxamin, benzamides, mundipropamide, cyanoimidazolesen, cyazofamide. Thiacarbamates, icaboxam, and amdoxims, siflufenamide, phenoxyquinolins, quinoxyf1, imidazoles, famidin, benzamides, flopicolide, zoxamide, representing different chemistries and mode of action. The majority of these have been developed for use against umicate pathogens. The main advantages of new generation compounds are ecologically safer and are required to be used at much lower rates than their earlier counterparts. A few of the lately developed fungicides have been registered for use in India and a good number of novel action fungicides are currently under evaluation. Azoxystrobins and phenamidin have been registered for use against grape downy mildew and potato blight. Prominent among those being tested against different diseases are mundipropamide, iprovalicarb, bentivlic, flopicolide, femoxazole, cyazofamide, pyraclostrobin and crisoxim methyl. These recently introduced fungicides to the market represent major advances in technology, potency against target diseases, selectivity, safety and rate reduction. However, they tend to have single-site modes of action which makes them potentially affected by target site resistance. Thus it is very important to proactively design and implement resistance management strategies and recommendations for new fungicide classes, as well as maintaining existing products. Swift development of resistance to strobilurins is now well documented and these are now categorized. 2258. Under high-risk fungicides. Their use has to be regulated and FRAG guidelines adopted so as to under high risk sustain their efficacy levels.